Settle in. There is no rush to what follows. The subject of this documentary is not mythology. It is not the future. It is the present, specific and documented. And it is strange enough that no embellishment is required. Less than 25% of the ocean floor has been mapped at a resolution high enough to detect features the size of a house. The rest, more than 3/4 of the largest continuous terrain on the surface of the Earth, exists in our records as smooth, averaged approximations derived from satellite measurements of sea surface height, translated through gravitational inference into rough estimates of what lies below. We do not know what is down there, not in the way that knowing means anything useful. We have outlines, we have profiles, we have models. We do not have maps. This is not a statement about ancient history. It is a statement about now, about this year, about the decade you are living in. The surface of the moon has been mapped in higher resolution than the floor of the Earth's oceans. The surface of Mars has been mapped in higher resolution than the floor of the Earth's oceans. Both of those bodies are an average of hundreds of millions of kilometers away from us. The ocean floor is an average of 3,688 meters below the surface of a medium we live on top of and have been sailing across for thousands of years. The gap between what we know about the surfaces of other worlds and what we know about the largest habitat on our own planet is not primarily a gap of technology. It is a gap of priority, funding, and the sheer physical difficulty of mapping the inside of an environment where the pressure at depth would collapse any ordinary container, and where radio waves, the carrier of almost all our long-distance sensing tools, cannot penetrate more than a few centimeters of seawater before being absorbed. Sound is the primary tool. And sound moves slowly, and collecting enough of it at enough locations to build a detailed map of a terrain covering 361 million square kilometers takes an enormous amount of time. The first systematic attempt to measure the depth of the ocean was made aboard the HMS Challenger, which departed Portsmouth, England, in December 1872 and returned in May 1876. The Challenger draped a weighted rope over the side of the ship at hundreds of locations across the Atlantic, Pacific, and Indian Oceans, let it sink until it reached the bottom, and measured how much rope had been paid out. This method, called sounding, worked. It was extraordinarily slow. A single measurement took hours. The Challenger made 492 deep soundings over the course of its 3.5-year voyage, covering 127,600 kilometers of ocean. The data it collected was transformative. It was also a point sample at 492 locations in an ocean that, at the scale of meaningful topographic resolution, requires tens of millions of measurements. Echo sounding, the use of sound pulses whose return time indicates depth, was developed in the early 20th century and became the standard tool for oceanographic depth measurement. The principle of echo sounding is simple. Send a pulse of sound downward, measure how long it takes to return, divide by the speed of sound in seawater, and you have the depth. The difficulty is that this gives you a single line of measurement directly beneath the ship, and covering the ocean with lines requires the ship to sail every line, which takes time proportional to the area being measured. Multi-beam sonar, which sends out a fan of sound pulses simultaneously and can map a swath of seafloor perhaps 10 times the water depth in width with each pass, became available in the 1970s and began transforming deep-sea mapping in earnest. But the math still does not favor speed. At a water depth of 4,000 meters, a multi-beam system can map a swath roughly 40 kilometers wide. The total area of seafloor below 200 meters depth is approximately 335 million square kilometers. To cover that area at the resolution available from ship-mounted multi-beam sonar would require a ship to sail lines spaced 40 kilometers apart across every ocean basin, which at a reasonable ship speed would take something on the order of 600 years of continuous effort. This is the physical constraint. The Seabed 2030 Initiative, a collaboration between the General Bathymetric Chart of the Oceans and the Nippon Foundation, set a goal of mapping the entire ocean floor at meaningful resolution by the year 2030. As of 2024, roughly 25% of the seafloor meets that goal. The remaining 75% is known only in the sense that gravity satellite data provides a rough profile of major features: ridges, trenches, large seamounts, at a resolution of approximately 5 kilometers. At that resolution, a submarine the length of a city block would be invisible. A mountain range 2 kilometers high might appear as a slight elevation in the data. The details, the canyon systems, the fault scarps, the volcanic fields, the rolling plains of sediment, remain unmapped. To understand what this means in human terms, consider the first time a human being actually saw the deep ocean with their own eyes. It happened in the Bermuda region in 1930, when a biologist named William Beebe and an engineer named Otis Barton lowered themselves into the Atlantic Ocean inside a steel sphere called the bathysphere. The bathysphere was attached to a cable and could not move under its own power. It had a single quartz window. On August 11, 1934, Beebe and Barton descended to 923 meters, a record that stood for more than a decade. Through that window, Beebe saw organisms he could not identify, creatures that produced bioluminescent light in patterns he had never imagined, things moving in the darkness at the edge of his lamp's reach that were clearly alive but whose identity he could not determine. He was the first vertebrate animal other than a diving seabird to see the mesopelagic zone with eyes. Beebe's accounts of what he saw through the bathysphere's window were questioned by some scientists at the time because they were so remarkable and because none of what he described could be collected and analyzed by the available technology. The creatures simply existed where he saw them, visible for a moment, and then gone back into the dark. He described things that later turned out to be real: the bioluminescent patterns of anglerfish, the counter-illumination of hatchetfish, and things that were never confirmed. The distinction between what he saw accurately and what he may have misinterpreted in the stress and novelty of the environment remains uncertain. What happened next, in terms of the deepest point of all, took another 26 years. In January 1960, a Swiss engineer named Jacques Piccard and a United States Navy lieutenant named Don Walsh descended into the Challenger Deep in the Mariana Trench aboard a bathyscaphe called the Trieste. The Trieste was not connected to a surface ship by cable. It was freediving, capable of moving under its own buoyancy, equipped with iron ballast that could be dropped to allow it to rise. It reached a depth subsequently measured at approximately 10,916 meters. Piccard and Walsh spent about 20 minutes on the bottom. They saw what they believed to be a flatfish on the sediment surface. They returned to the surface. No human being returned to the Challenger Deep for more than 50 years. The interval between 1960 and 2012, when filmmaker James Cameron made a solo dive to the Challenger Deep in a purpose-built submersible called the Deep Sea Challenger, represents 52 years during which no human visited the deepest point on Earth. In those 52 years, humans landed on the moon 6 times, sent spacecraft to every planet in the solar system, built the International Space Station, developed the Internet, sequenced the human genome, and mapped the surface of Mars at resolutions better than a meter per pixel. During those same 52 years, the deepest point on Earth was visited by machines occasionally, and by no human at all. The asymmetry is complete, and it reflects genuine choices about where human curiosity and human resources are directed. The Challenger Deep is not the only unvisited deep. Every major Hadal Trench, the Tonga Trench, the Philippine Trench, the Kermadec Trench, the Japan Trench, the Puerto Rico Trench, has been visited at its deepest point a handful of times or never. The 5-kilometer deep basins of the Central Pacific, the South Atlantic, and the Indian Ocean contain almost no direct observations at all. The Mid-Ocean Ridge System, the longest mountain range on Earth at 60,000 kilometers, is known in geological outline from echo sounding and in biological detail from perhaps a few hundred localized sampling efforts. The vast majority of the ridge has not been visited at all. The satellite data that gives us our rough picture of the global seafloor reveals, when analyzed carefully, that there are approximately 100,000 seamounts in the ocean, defined as underwater mountains rising more than 1,000 meters from the surrounding floor. Of these, fewer than 500 have been directly sampled or imaged by any research vessel. The rest exist as bumps in the satellite gravity data. Each seamount is an isolated elevated habitat in the deep ocean, with its own current regime, its own depth range, its own substrate type. The organisms that live on an isolated seamount in the middle of the South Pacific may have evolved in near-complete isolation from the organisms on the next seamount 500 kilometers away. They may be the same species. They may be entirely different. We do not know because we have not looked. What this means scientifically is not a matter of speculation. It means that the ocean floor contains geological features that have never been observed. It means that organisms live in environments where no camera has ever arrived. It means that processes are occurring in places where no sensor has been deployed. The deep ocean is not empty. It is full. We are simply not there to see most of it. The question that follows from this reality is, what happens when we do go? The answer, repeated consistently across the history of deep-sea exploration, is that we find things that were not expected. The hydrothermal vents discovered in 1977 by the crew of the submersible Alvin near the Galapagos Rift were not predicted by any oceanographic model at the time. The cold seeps discovered in the Gulf of Mexico in 1984 were not predicted. The Lost City hydrothermal field, discovered in 2000 on the Mid-Atlantic Ridge, was not predicted. Every major biological community type in the deep ocean was discovered as a surprise, not as a confirmation of something previously theorized. Consider specifically what happened in 1977. The Alvin submersible was conducting geological surveys of the seafloor near the Galapagos Spreading Center, looking at the ridge system where the Pacific and Nazca tectonic plates are slowly moving apart. The researchers expected to see basalt, sediment, and relatively sparse marine life adapted to the usual deep-sea conditions of cold temperature and limited food supply. What they found instead was a community of organisms unlike anything previously documented. Tube worms 2.5 meters long in white tubes anchored to the rock, clams the size of dinner plates, dense mats of bacteria coating every surface, crabs and fish and shrimp moving through the warm water rising from cracks in the seafloor. The water temperature near the vents was several hundred degrees Celsius, emerging from the Earth's interior as a consequence of the volcanic activity at the spreading center. The organisms living around these vents were not obtaining energy from sunlight, directly or indirectly. They were obtaining energy from the chemical gradient between the hot, mineral-rich vent fluid and the cold, oxygen-rich seawater. The bacteria at the base of the food web were oxidizing hydrogen sulfide, a compound toxic to most life, and using the energy released to fix carbon dioxide into organic compounds. This process, chemosynthesis, was known in the laboratory but had not been observed as the basis of a large, complex ecosystem in nature. The 1977 discovery established that life on Earth did not require sunlight. It established that the deep ocean, rather than being a barren place dependent on scraps of organic material falling from the surface, could support dense, diverse communities powered by the Earth's own interior heat. This was not a minor discovery. It changed the fundamental understanding of where life can exist and how it can be sustained. It has implications for the search for life on other planets and moons, particularly the ice-covered oceans of Jupiter's moon Europa and Saturn's moon Enceladus, where similar hydrothermal activity may occur at the base of a subsurface ocean. The discovery was made accidentally during a survey that was looking for something else entirely. The ocean does not announce what it contains. It holds its inventory quietly. And every time something goes down and looks, the inventory is larger and stranger than the record suggested before the looking. The discovery of chemosynthetic communities pointed to something broader about the ocean, that it contains entire modes of existence that were invisible to science until someone physically arrived at the right location at the right time with the right equipment. That same principle applies not only to what lives in the deep ocean, but to what the deep ocean sounds like and what is making the sounds. The ocean is not quiet. It is, in fact, the noisiest environment on the planet when measured across the full frequency range of sound that water can carry. The thermal noise of water molecules in constant motion, the sounds of breaking waves transmitted downward from the surface, the calls of whales and fish and shrimp and every other sound-producing organism, the low-frequency rumbles of undersea earthquakes and volcanic eruptions, the creak and groan of tectonic plates shifting against each other. All of this travels through the ocean simultaneously, in all directions, at a speed of approximately 1,500 meters per second, which is roughly 4 times faster than sound moves through air. The deep ocean has a structural feature that makes it an especially effective acoustic environment. At a depth of approximately 600 to 1,200 meters in most parts of the ocean, the combination of temperature and pressure creates a zone where the speed of sound reaches a local minimum. Sound traveling through water naturally bends toward zones of lower sound speed. This means that any sound produced at or near this depth zone tends to be refracted back toward it from both above and below, trapping it in a horizontal channel that carries it with very little spreading loss over enormous horizontal distances. This channel is called the SOFAR channel, sound fixing and ranging channel, and sounds produced within it can travel thousands of kilometers across entire ocean basins before dissipating below the threshold of detectability. The military implications of the SOFAR channel were recognized during the Second World War. A system called SOFAR was developed as a rescue tool, Downed pilots could set off a small explosive charge at SOFAR channel depth, and hydrophone arrays could triangulate the position of the explosion from the sound's arrival times at widely spaced listening stations. The same principle was later scaled up into a network of bottom-mounted hydrophones called SOSUS, Sound Surveillance System, deployed by the United States Navy during the Cold War to track Soviet submarines across the North Atlantic and North Pacific. By the late 1980s and early 1990s, the Cold War was ending and SOSUS was partially declassified and made available to civilian scientists. What civilian scientists found when they began listening to the data from SOSUS and from similar monitoring systems around the world was extraordinary. The ocean was full of sounds that had no identified source. In 1997, the National Oceanic and Atmospheric Administration, NOAA, recorded an ultra-low frequency sound in the South Pacific Ocean that was unlike any sound in their reference catalog. The sound rose rapidly in frequency over a period of about 1 minute, then fell away. It was extraordinarily loud at its source, estimated to have originated somewhere in the southern Pacific, at a level that would have required either an enormous natural event or an organism larger than any animal known to produce sound in that frequency range. The source was never identified. NOAA named the sound the bloop. The bloop was detected on multiple hydrophones simultaneously, allowing its approximate origin to be triangulated to a region of the southern Pacific Ocean near 50 degrees south latitude, 100 degrees west longitude. This region is remote, deep, and largely unmonitored. The frequency profile of the bloop, the way it rose and fell in pitch, showed characteristics similar to biological sounds, specifically the kind of rapid frequency sweeps produced by large marine mammals. But no known marine mammal produces sounds in that frequency range and at that apparent source level. A blue whale, the loudest animal known, produces sounds in the 10 to 40 Hz range at source levels of around 180 decibels, referenced to 1 micropascal. The bloop's apparent source level and frequency profile exceeded what any known animal could produce. One hypothesis proposed in subsequent years is that the bloop was produced by an ice quake, the fracturing and calving of Antarctic ice, rather than by any living organism. The frequency profile of ice quakes can in some cases resemble that of biological sounds, and the southern Pacific near the Antarctic convergence zone is an area where large ice events occur. NOAA's current position, which has evolved over the years since the original recording, leans toward a natural geophysical origin. But the sound has not been definitively identified, and no subsequent recording has been made that allows a final determination. The bloop is the most famous of the unidentified ocean sounds, but it is not the only one. In 1991, NOAA monitoring systems began detecting a long-duration signal in the equatorial Pacific, a sound they called upsweep because it consisted of narrow-band upsweeping tones repeated over minutes to hours. Upsweep was detected seasonally with higher intensity during Northern Hemisphere spring and autumn, and its apparent source was localized to an area of seamount activity in the south-central Pacific. The seasonal variation suggested a possible volcanic origin, but upsweep does not match the acoustic signature of any known type of volcanic activity precisely. It has been recorded regularly since 1991 and remains unexplained. In May 1997, NOAA detected a sound that lasted about 15 seconds, with a profile they described as resembling the sound of a train passing, a regular series of intensity peaks. This sound, called train, was recorded on multiple hydrophone arrays in the Pacific and localized to a region near Antarctica. Its origin is not established. In the same year, NOAA recorded another anomalous sound they called slow down, a sound that decreased steadily in frequency over about 7 minutes, with properties unlike any volcanic or biological source previously identified. Slowdown has been detected multiple times since 1997, originating from roughly the same area of the South Pacific each time, and its source remains unexplained. The sound, called Julia, detected in March 1999 on the Eastern Pacific Monitoring Array, lasted about 14.8 seconds and was exceptionally loud. The frequency profile of Julia was unusual. It did not match any known geological or biological source precisely. Its localized origin appeared to be somewhere in the southern Pacific near the Antarctic convergence zone. No subsequent analysis has produced a confident identification. These named sounds, bloop, upsweep, train, slow down, Julia, represent the cases that were notable enough to receive designations in NOAA's recording catalogs. They are not all the anomalous sounds detected in those archives. The archives contain many sounds that were recorded, analyzed briefly, found to match nothing in the reference catalog, and filed without a confident identification. The number of anomalous sounds in the full dataset of ocean acoustic monitoring is not published, but scientists who work with the data indicate that genuinely puzzling recordings are not rare. For 4 decades, a series of sounds was recorded in Antarctic waters that did not match any cataloged biological source. The sounds were repetitive, patterned, and occurred at frequencies consistent with large marine mammals, but did not match the known call patterns of any identified Antarctic species. They were detected widely enough to have been colloquially named the bioduck, because some researchers thought they resembled the sound a duck might make if greatly slowed down and played at very low frequency. The bioduck was first noted in acoustic recordings from the 1970s and continued to be recorded for decades. It was heard by submarine crews operating in Antarctic waters, who sometimes reported hearing unusual repetitive sounds through the hull. The source was genuinely unknown for 40 years. In 2014, a team of researchers from the Australian Antarctic Division published a study in the journal Biology Letters that finally identified the source of the bioduck. They had attached acoustic recording tags to Antarctic minke whales, Balanoptera bonarensis, and found that the tags produced recordings that matched the bioduck exactly. The sound was being produced by Antarctic minke whales during feeding dives, likely as part of their foraging or communication behavior. A sound that had been recorded for 40 years and that stumped researchers for 4 decades turned out to come from one of the most abundant whale species in the Southern Ocean. The identification of the bioduck is worth sitting with for a moment. Antarctic minke whales are not rare or unknown. They are among the best-studied whale species in the Southern Ocean, with a population estimated in the hundreds of thousands. They have been the subject of intensive research for decades, including research specifically focused on their acoustics, and yet the sound they produced during a specific behavior, a repetitive, distinctive, frequently recorded sound, went unattributed to them for 40 years. The sound was being heard by submarines and recorded by fixed hydrophones and analyzed by researchers throughout this period. No one connected it to the animal until a tag was placed directly on the animal, and the sound was recorded simultaneously with the animal's behavior. The lesson is not that scientists are careless. The lesson is that the ocean is large and acoustic identification of biological sources is genuinely difficult. Most ocean acoustic research is done by analyzing recordings from fixed hydrophones, without any ability to simultaneously observe what is happening at the location of the sound. Attaching a tag to an animal requires first catching or approaching the animal, which is difficult in the open ocean and especially difficult in Antarctic waters. The gap between the audio record and the biological identification can persist for a very long time. What the bioduck discovery demonstrates is that we can have detailed, consistent, high-quality recordings of an ocean phenomenon for 4 decades and still not know what is producing it. The bloop, the upsweep, the train, Julia and slowdown all remain without a definitive source identification. Each of them has a working hypothesis: icequakes, volcanic activity, or unknown biological sources in various combinations. But none has been definitively solved, and these are only the sounds that were interesting enough to receive names. The full acoustic environment of the deep ocean, at the frequencies and depths accessible to the SOFAR channel, remains mostly uncharacterized. Consider what it means that the ocean carries sound from one end of a basin to another. A sound produced in the southern Pacific can be detected in the northern Pacific hours later. A whale calling in the North Atlantic may be heard by hydrophones in the Mediterranean. The acoustic environment of the deep ocean is not local. It is global. Everything producing sound at SOFAR channel depth, anywhere in the ocean, is theoretically detectable anywhere else in the ocean with the right equipment. We have the equipment. We are recording constantly, and we are still regularly encountering sounds we cannot explain. The ocean speaks. We are only beginning to understand the language. The acoustic mysteries of the deep ocean represent one layer of the unknown. We can detect signals, but cannot always determine their source. There is a deeper layer of the unknown, one that became apparent only in the 1990s, and that has not yet been fully incorporated into popular understanding of the ocean's biology. The ocean has a floor. Below the floor, there is rock. Rock is not alive. This has been the operating assumption of marine biology for most of its history as a discipline, and it is largely correct. But in 1994, a paper published by John Parks and colleagues in the journal Nature reported the results of a systematic study of deep-sea sediment cores from the Pacific and Atlantic Oceans. The sediment cores had been drilled by the Ocean Drilling Program, which routinely penetrated hundreds of meters into the seafloor as part of geological and paleoceanographic research. Parks and his team analyzed the microbiology of these cores, looking for signs of living organisms at depth. They found them. Living bacteria and archaea, another domain of single-celled life, distinct from bacteria and from eukaryotes, were present in sediment hundreds of meters below the seafloor surface, at depths where the sediment had been accumulating undisturbed for millions of years. The organisms were metabolizing slowly, living in chemical environments with very little energy available, surviving on traces of organic carbon buried in the sediment or on chemical reactions between minerals and water. They were alive, and they were present everywhere the cores were analyzed. Subsequent research extended the discovery. In 2012, the Integrated Ocean Drilling Program recovered sediment cores that contained living organisms at depths of more than 2,500 meters below the seafloor surface. Sediment that was 86 million years old. The organisms were alive, growing and dividing, though at rates so slow they would divide perhaps once every 10,000 years. They were surviving in conditions of near-zero energy availability, in sediment that had been compressing and lithifying around them for tens of millions of years, in the complete absence of light, in temperatures warm enough from the geothermal gradient below but low enough at the sediment surface to be near freezing. This community of organisms in the sediment below the seafloor, the deep biosphere, is now estimated to contain a biomass comparable to the total biomass of all surface life on Earth combined. That estimate carries enormous uncertainty, because the deep biosphere has been sampled at only a tiny fraction of the locations where it exists, and because the density and metabolic rate of the organisms varies enormously with depth, sediment type, and available chemical energy. But the estimate, even if off by a factor of 10, means that the largest community of living organisms on Earth by total mass may be the one living beneath the floor of the ocean, invisible, slow, alien in its metabolism, unknown in most of its biology. The organisms of the deep biosphere are not the same as the organisms of the deep sea floor. They are not eating marine snow or tube worms or whale falls. They are, in many cases, obtaining energy from reactions between water and rock. The serpentinization of olivine and other minerals in oceanic crust produces hydrogen, which can serve as an energy source for certain archaea and bacteria. They are living in geological time rather than biological time, completing life cycles that span what we would consider geological epochs. The discovery of the deep biosphere is relevant here, not just as a scientific fact, but as a demonstration of the principle the ocean keeps repeating, that entire categories of life can exist in environments that were assumed to be sterile or uninhabitable, completely outside the framework of any prior theory, waiting to be found by the first person who actually looked. In the same category of surprising discovery belongs the Lost City hydrothermal vent field, found in December 2000 by a research expedition using the submersible Alvin on the Mid-Atlantic Ridge. The field was discovered during a research cruise that had another primary objective. The crew of the Alvin, descending to survey a geological feature called the Atlantis Massif, found instead a field of extraordinarily tall carbonate chimneys. The tallest one, named Poseidon, reaches approximately 60 meters above the seafloor, the height of a 20-story building, producing warm, alkaline fluid that was the result of serpentinization of the ultramafic rock of the oceanic crust. Lost City was immediately recognized as different in chemistry and biological character from the black smoker hydrothermal vents known since 1977. Where black smokers produce hot, acidic, metal-rich fluid driven by magmatic heat close to the seafloor, Lost City produces cooler, alkaline, hydrogen-rich fluid driven by the exothermic reaction between seawater and the olivine-rich rock of the oceanic mantle. The biological community at Lost City is dense with archaea, methanogens, and other organisms that use hydrogen and carbon dioxide as energy sources, and the carbonate chimneys themselves are inhabited by large mats of microbial life. Lost City is also distinctive for its permanence. Black smokers tend to be episodic, associated with volcanic events that create them and then kill them over periods of decades. Lost City has been active, based on radiometric dating of its carbonate structures, for at least 30,000 years, and possibly much longer. It may represent a type of hydrothermal activity that has been continuous since the formation of the oceanic crust at this location, meaning it could have been active for millions of years. It exists in a location that, before the 2000 expedition, had not been specifically targeted for vent surveys. The chemistry of Lost City has attracted attention from researchers interested in the origin of life. The serpentinization reaction produces not only hydrogen, but also small organic molecules, including simple amino acids and fatty acids, as byproducts. The alkaline, warm, hydrogen-rich environment of Lost City has been proposed as analogous to the conditions in which the first self-replicating molecules may have formed on early Earth. This remains a hypothesis, one of several competing theories about the origin of life, but the physical chemistry of the site makes it plausible in a way that no prior known environment did. The discovery of Lost City was not simply a new biological community. It was a new window into deep Earth chemistry, and potentially into the conditions that made life possible in the first place. The deep ocean contains other kinds of biological mystery that are less geological and more anatomical. The Vampiroteuthis infernalis, whose name translates roughly as the vampire squid from hell, a name coined by its scientific describer in 1903, is an animal that defies easy classification. It is not a squid in the true sense. It is not an octopus. It belongs to its own order, Vampiromorphida, which contains only one living species and which sits phylogenetically between the squids and the octopuses without being clearly one or the other. Vampiroteuthis lives primarily in the oxygen minimum zone, between about 600 and 900 meters depth in tropical and subtropical waters worldwide. The oxygen minimum zone is the depth band where the concentration of dissolved oxygen in the water reaches its lowest point, a consequence of the intense biological decomposition of organic material sinking from the surface above, which consumes oxygen, combined with the limited replenishment of oxygen at these intermediate depths, where mixing with surface water is slow. Many organisms cannot survive in the oxygen minimum zone because they require more oxygen than is available. Vampiroteuthis survives because of an unusually efficient blood pigment. Its hemocyanin, the copper-based oxygen carrier that makes its blood blue rather than red, has an exceptionally high affinity for oxygen, allowing the animal to extract oxygen from water where most other animals cannot. The animal is small, reaching about 30 centimeters in mantle length in adults. It is dark red-brown in color with large blue eyes, the largest eyes relative to body size of any animal, and a web of skin connecting its 8 arms. It can retract its arms back over its body when threatened, presenting the spines on the inner surface of the webbing as a defense. It does not have ink sacs, which squids use for defense. Instead, it has glands at the tips of 2 specialized appendages that can produce a cloud of bioluminescent mucus, confusing a predator in the dark water. Everything about the anatomy of Vampiroteuthis reflects its specific adaptation to an environment that most animals avoid. What Vampiroteuthis eats remained largely unknown until 2012, when researchers from Mberi published a study based on analysis of the contents of the mucus strands the animal uses to catch food. The diet turns out to be almost entirely marine snow, the aggregated organic detritus falling from the surface above. Vampiroteuthis is not a predator in any conventional sense. It is a detritivore, a scavenger of the organic rain that falls continuously through the water column, catching it on long, sticky filaments and consuming it in a way that no other cephalopod does. This feeding strategy was not predicted from the animal's anatomy or from any behavioral observation before the 2012 analysis. Vampiroteuthis looks like a predator. It is not. The barreli fish of the family Opisthophroctidae is a different kind of anatomical mystery. The name barreli refers to the tubular eyes that characterize these fish, cylindrical structures pointing upward from within the head, with the retina at the bottom of each cylinder receiving light focused by the lens at the top. The tubular eyes allow an extremely wide aperture relative to the retina size, providing exceptional sensitivity in low light conditions. And they are pointed upward because the fish's primary task is to detect the silhouettes of organisms above it against the faint residual light from the surface, the same counter-illumination problem that hatchetfish solve by producing light downward. For most of the 20th century, what was known about barreli fish anatomy came from preserved specimens, which were collected in trawl nets and fixed in formalin or alcohol for museum collections. In preserved specimens, the tissue covering the top of the head is opaque. It desiccates and loses transparency during preservation. The eyes, in preserved specimens, appear to be fixed in their upward orientation and surrounded by white, opaque tissue. In 2008, researchers at Mberi published footage collected by their ROVs of living barrel-eye fish in their natural habitat, in the mesopelagic zone off the California coast. The footage revealed two things that the preserved specimens had never shown. First, the top of the head was transparent in the living animal, a fluid-filled dome of clear tissue through which the tubular eyes were visible from the outside, like two green cylinders inside a glass case. Second, the eyes were not fixed in their upward orientation. They could rotate forward, so that the fish could look directly ahead when pursuing prey, and rotate back to their default upward orientation when the fish returned to scanning mode. The combination of forward-looking eyes with extremely wide aperture lenses allows the fish to detect and pursue small organisms at extremely close range in very dim light, then return to scanning the water column above for silhouettes. Before 2008, none of this was known. The barrel-eye fish was a described species with a scientific name and a museum record going back to the 19th century. Its anatomy had been studied and published. Anatomists who examined preserved specimens noted the white, opaque dome of tissue on the head without understanding what it represented. The function of the eye rotation mechanism could not be inferred from preserved material because the muscles that control the rotation lose their function after death and fixation. The living animal was completely different from the preserved record. This is not an isolated case. It is a pattern. The living animal in its natural environment consistently reveals features that the preserved specimen cannot. For every barrel eye, for every Vampiroteuthis, for every Osidax worm in a whalebone, organisms discovered alive in their habitat, revealing behaviors and structures that the dead or captive specimen could never have shown. There are organisms in museum jars whose full living biology is still unknown for exactly the same reasons. The record of deep-sea biology is a record of dead things. The living ocean is still, in most of its volume, unobserved. The pattern of discovery, arriving somewhere new or arriving somewhere familiar with new instruments and finding something that was entirely outside the framework of existing knowledge, repeats across every dimension of deep-sea research. The acoustic record is full of unidentified sounds. The biological record is full of preserved specimens whose living behavior is unknown. The geological record is full of features that have been detected by sonar but never approached by any camera. And the oceanographic record, the understanding of how the deep ocean functions as a physical and chemical system, is also, in specific important ways, incomplete. Oxygen minimum zones are one of the best documented examples of a large-scale oceanographic phenomenon that was poorly understood until the mid-20th century and whose full implications are still being worked out. An oxygen minimum zone is a depth band within the ocean, typically between 200 and 1,000 meters, where dissolved oxygen concentrations fall to levels that are very low or, in some regions, effectively zero. These zones form because of a combination of factors. Organic matter sinking from the productive surface ocean is broken down by bacterial decomposition at these intermediate depths, consuming dissolved oxygen in the process. While the replenishment of oxygen from the surface is slow because these depths are below the wind-mixed surface layer and do not directly communicate with the atmosphere. The largest oxygen minimum zones in the global ocean are found in the eastern tropical Pacific and the Arabian Sea, where particularly productive surface water supplies abundant sinking organic material and where the water circulation patterns slow the delivery of oxygenated water from depth. In these regions, the OMZ can extend from as shallow as 50 meters to as deep as 1,000 meters and cover hundreds of thousands of square kilometers of ocean volume. The oxygen concentrations within these zones can be below 0.5 milliliters of oxygen per liter of water, compared to surface water values of around 8 milliliters per liter. At these concentrations, most fish and most invertebrates cannot survive, and the biological community within the OMZ is dominated by specialized organisms adapted to low oxygen. The scientific history of oxygen minimum zones is in part a history of how long it took oceanographers to recognize that large regions of the ocean were persistently hypoxic, that this was not an anomaly or a local condition but a structural feature of ocean chemistry repeated across basin scales. The Arabian Sea OMZ covers approximately 10 million square kilometers of ocean volume. It is not subtle, but measuring dissolved oxygen at depth across the open ocean requires deploying instruments over enormous areas, and systematic measurement programs only became practical as ships and instruments became more capable through the 20th century. What has become clear since the 1990s is that oxygen minimum zones are expanding. Dissolved oxygen concentrations in the global ocean have been declining measurably over the past 50 years, and the vertical extent of OMZs, how thick the low oxygen layer is at any given location, has been increasing. A 2017 study published in the journal Science, analyzing data from multiple oceanographic observing programs, estimated that the volume of the global ocean with oxygen concentrations below 70 micromoles per kilogram, a threshold below which many marine organisms experience physiological stress, had increased by approximately 4.5 million cubic kilometers between 1960 and 2010. That is a volume roughly 50 times the total volume of the Mediterranean Sea, added to the globally hypoxic ocean in 50 years. The biological consequences of expanding OMZs are complex and not fully understood. As the hypoxic volume expands upward into shallower water, it compresses the habitat available to organisms that require higher oxygen levels, forcing them into a narrower range of depths near the surface. This compression of habitat can increase the vulnerability of these organisms to surface predators and fishing pressure. At the same time, some organisms that are adapted to OMZ conditions may benefit from an expanding OMZ, and the overall reorganization of the midwater ecosystem that results from changing oxygen distributions is not fully characterized. The organisms that live within oxygen minimum zones, by contrast, are among the most bizarre in the deep ocean. Vampiroteuthis infernalis, already described, is one. Many of the microbial residents of OMZs use chemical pathways that do not involve oxygen at all: denitrification, reduction of sulfate, oxidation of ammonia through a process called anammox. And these pathways produce gases, including nitrogen and nitrous oxide, that have implications for the global chemistry of the atmosphere. The OMZ microbiology is an active area of research precisely because the microbes there are doing things with global-scale consequences in a poorly understood way. In September 2005, the crew of the SS Lima, a British merchant vessel sailing through the Indian Ocean northwest of Australia, reported seeing a vast area of brightly glowing water at night. The glow covered an area they estimated at about 250 square kilometers and was bright enough to read a book by from the deck of the ship. They reported the observation, and the crew were initially uncertain what they had witnessed. Glowing water is not unprecedented. Bioluminescence in the surface ocean is common and has been observed since ancient times by sailors. But the scale and intensity of what the Lima encountered was remarkable. A team of researchers examining satellite data from the Defense Meteorological Satellite Program subsequently located an area of bright bioluminescence in the Indian Ocean at approximately the time and location of the Lima's encounter. The satellite detected a glowing patch of ocean approximately 15,000 square kilometers in area, about the size of the state of Connecticut, that was visible from space. The researchers published their analysis in 2005, providing the first satellite-documented case of what sailors have historically called the Milky Sea. The Milky Sea phenomenon, also called Sea of Milk, or Mare Lateo in Italian, has been recorded in sailors' logs and historical accounts for hundreds of years, primarily from the Indian Ocean and the Arabian Sea. Sailors described seeing a vast, uniform glow across the sea surface at night, not the flickering bioluminescence produced by disturbed organisms, but a steady, diffuse light covering the horizon in all directions. The phenomenon was so striking that it appeared in literature, including Jules Verne's 20,000 Leagues Under the Sea, and was sometimes used as a navigational reference. It was also consistently dismissed by scientists who had not witnessed it directly as an optical illusion or an exaggeration. The 2005 satellite observation validated the historical accounts. The Milky Sea is real. It is caused by an extraordinary concentration of bioluminescent bacteria, specifically a species called Noctiluca scintillans, combined with a bloom of the bacterium Vibrio harveyi and related species, which produce a steady bioluminescent glow rather than the flash-on-demand glow produced by many larger organisms. A Milky Sea event requires an enormous mass of bacteria covering a large area of ocean surface to produce the visible effect. The conditions that produce such blooms, warm water temperatures, specific nutrient conditions, calm seas that allow the bacteria to concentrate, are not common, but are not rare either, particularly in the western Indian Ocean. The immediate question raised by the validated Milky Sea phenomenon is, How many other historical sailor accounts of anomalous ocean observations have been similarly dismissed as exaggeration and are similarly real? Sailors' logs from the age of sail contain numerous accounts of strange ocean observations, unusual colors, strange sounds, anomalous currents, things seen at night that could not be explained by daylight examination of the same area. The scientific literature has generally treated these accounts with skepticism, accepting them only when corroborating evidence exists. But the Milky Sea case suggests that the bar of skepticism may sometimes have been set too high. Observers at sea were often careful and accurate. What they saw was sometimes real. The deep ocean's role in the global climate system represents another dimension of the unknown. The ocean stores heat. It absorbs carbon dioxide from the atmosphere. It distributes both heat and carbon to distant locations through the movement of its water. These are well-established facts at a general level. The specific mechanisms, the rates, and the biological contributions to these processes are, in key respects, still being worked out. The biological carbon pump is the process by which organisms in the surface ocean fix atmospheric carbon dioxide into organic compounds through photosynthesis, and some fraction of those compounds eventually sinks to depth and is sequestered there, away from the atmosphere, for timescales from decades to millennia. Phytoplankton at the surface produce organic carbon. Some of that carbon is consumed by zooplankton. Some is packaged into fecal pellets that sink faster than free cells. Some aggregates into marine snow. Some is carried to depth by organisms in the deep scattering layer that feed near the surface at night and carry the carbon they have consumed down to their daytime depths. All of this represents a downward flux of carbon that removes it from the surface ocean and atmospheric exchange. The magnitude of the biological carbon pump, how many gigatons of carbon it moves from the surface to the deep ocean per year, has been estimated using a variety of methods, and the estimates have consistently disagreed with each other by factors of 2 or 3. Part of the problem is the mesopelagic zone. Organisms living in the mesopelagic consume some of the sinking organic material before it reaches the deep ocean. How much they consume and how much passes through to the abyssal floor depends on the composition and abundance of the mesopelagic community, which, as previously discussed, is not well characterized. Improving the estimate of the biological carbon pump's magnitude requires better knowledge of the mesopelagic community's abundance and metabolic rate, which requires better surveys, which requires more ship time and more autonomous instruments. The deep ocean, in its chemical and physical functioning, is not a passive reservoir. It is an active participant in the global cycling of carbon, nitrogen, oxygen, and other elements, and that participation is mediated by the organisms it contains. The organisms it contains are mostly unknown. The ocean's participation in the global chemical cycles is consequently understood only in its broad outlines, not in the detail required to predict how it will respond to changing conditions at the surface. This is not a failure of ocean science. It is an accurate description of where ocean science currently stands. The detail is genuinely not there yet. There is a scientific tradition in which the end of a field of inquiry is in sight, where the major discoveries have been made, where the remaining work is detail and confirmation, where the fundamental picture is correct even if imprecise in some specifics. Particle physics entered this mode after the confirmation of the Higgs boson in 2012. Classical mechanics entered it after Newton. Descriptive anatomy of the human body was largely complete by the early 20th century, though molecular biology opened an entirely new layer of complexity beneath it. Deep sea biology is not in this tradition. It is not approaching completion. The discovery rate of new species, new ecological communities, new physiological capabilities, new behaviors, new geological contexts has not decreased. If anything, the instruments available for deep-sea research have improved enough in the past 2 decades that the rate of discovery is increasing, not because there is more to discover than there was before, but because we are now better equipped to find it. Every major expedition to a previously unvisited or undersampled location returns with species new to science. Every major application of environmental DNA sampling to the water column reveals genetic sequences that match no described organism. The ocean is not running out of surprises. The history of what we thought we knew about the deep ocean and how consistently we turned out to be wrong is instructive. Before 1977, it was believed that all life in the deep ocean ultimately depended on the surface productivity of the ocean above. That the food chain of the deep sea was a long, attenuated version of the food chain of the surface, sustained by the slow rain of organic material drifting down from the productive layers above. This was a reasonable hypothesis, consistent with everything that was known. It turned out to be wrong in a fundamental way. Entire ecosystems exist in the deep ocean that do not depend on surface productivity at all, that are sustained by chemical energy from the Earth's interior. Not wrong in a detail. Wrong in the foundational assumption about where the energy comes from. Before 1994, it was believed that the rock below the seafloor was, at most, a substrate for benthic organisms living on the seafloor surface. The sediment below the seafloor was not considered a habitat. It turned out to be among the largest habitats on Earth, containing a community of organisms whose total biomass may rival that of all surface life combined. Not wrong in a detail. Wrong about whether the habitat existed at all. Before 2000, the alkaline, serpentinization-driven hydrothermal system represented by Lost City was a theoretical possibility, but had never been observed. The existence of such systems, and their particular biological and chemical character, was not incorporated into any ecological or geochemical model of the ocean floor. The discovery of Lost City did not simply add a new location to an existing category. It defined a new category. Before 2008, the anatomy of the barrel-eye fish, as understood from preserved specimens, was incomplete in a way that no one knew it was incomplete. The transparent dome, the mobile eyes, the full functional picture of how the animal uses its unusual anatomy, all of this was invisible in the preserved record. The animal had been known to science for over a century. Its living biology was discovered in the 21st century. This pattern, confident prior knowledge repeatedly revealed as incomplete or wrong, is the deepest argument for the claim in this documentary's title. The deep sea is not almost known. It is not mostly known with some remaining details. It is known at the level of broad categories and rough outlines, with the specific content of those categories and the detail within those outlines almost entirely unknown. The surprises are not getting rarer. They are getting better documented as the instruments improve, which is not the same thing as getting rarer. The pharmaceutical implications of the deep ocean are a specific case in point. The deep-sea organisms that have been collected and analyzed for bioactive compounds have already produced several compounds of medical significance. The anti-tumor compound trabectedin, marketed as Yondelis, was originally isolated from a sea squirt, a tunicate, found in shallow Caribbean waters. But related compounds with potential medical utility have been found in deep-sea tunicates and sponges. A class of compounds called polyketides, produced by deep-sea fungi and bacteria living in sediments, has shown antimicrobial and anti-cancer activity in laboratory screens. A compound isolated from a deep-sea sponge of the genus Discodermia, discodermalide, has demonstrated potent anti-tumor activity and has been studied as a potential cancer treatment. The deep-sea sponges are of particular interest for pharmacological research because they have been living in environments where they cannot move to escape predators or chemical threats, and they have consequently evolved an enormous chemical arsenal, secondary metabolites whose biological function is defense against predation or competition. These chemicals are, by selection, bioactive. They interfere with the biological processes of other organisms. That same interference is often useful in medicine, where drugs work by specifically disrupting biological processes, blocking enzymes, disrupting cell membranes, inhibiting DNA replication in pathological cells or organisms. The fraction of deep-sea organisms that have been collected and analyzed for bioactive compounds is extremely small. The number of deep-sea species that have been subjected to systematic chemical screening for medically useful compounds is in the hundreds. In an ocean where the total number of species is estimated in the millions and the vast majority have never been collected. Every organism in the deep ocean that has not yet been described is a potential source of compounds that have not yet been identified. This is not a theoretical possibility. The precedent is established. The question is only how many discoveries are waiting in the fraction of the ocean that has not been examined. The future of deep-sea exploration will be shaped by two technologies that are currently at the beginning of their impact. The first is autonomous underwater vehicles, AUVs, capable of operating without a tether to a surface ship, navigating by acoustic positioning or pre-programmed routes, and operating for extended periods at depths of thousands of meters. Several programs are developing AUV fleets designed to carry out systematic survey work across large areas of the seafloor that ship-based ROVs cannot practically cover. An AUV can be deployed from a ship, complete a survey of several hundred square kilometers of seafloor, and return to be recovered, while the ship sails to the next deployment location. multiple AUVs can be deployed simultaneously, multiplying the coverage rate. The second technology is environmental DNA sampling at scale. eDNA, the collection and sequencing of genetic material shed by organisms into the surrounding water, is already producing biological inventories of deep-sea water columns at locations and depths where no biological sampling program has previously operated. As the cost of DNA sequencing continues to decline, it becomes practical to filter and sequence water samples from thousands of locations across the global ocean, producing a global-scale genetic atlas of what lives where. This atlas will not tell us everything. eDNA cannot reveal behavior, body form, ecological relationships, or biochemistry directly, but it will tell us where the organisms are, roughly how diverse the community is at each location, and which locations are most in need of targeted sampling with more detailed methods. Together, these technologies have the potential to substantially change the picture of deep-sea biodiversity within the next 2 to 3 decades. The combination of systematic AUV surveys, global-scale eDNA sampling, and continued targeted investigation of specific habitat types should produce a clearer view of what the deep ocean contains and where different communities live than has ever existed before. But there is a constraint that technology cannot easily overcome: the description and analysis of what is found. Environmental DNA surveys can detect the presence of an unknown organism and estimate its abundance from the concentration of its genetic material in the water. They cannot tell us what the organism looks like, what it eats, what eats it, how it reproduces, or what ecological role it plays. All of those questions require either collecting the organism, which requires a net or suction sampler or manipulator arm delivered to the right location, or observing it in situ with a camera and recording its behavior. Collection and in situ observation are slow, expensive, and dependent on ship time and submersible time, which remain limited even as autonomous platforms expand the geographic reach of deep-sea research. The consequence is that the rate of discovery of new species from eDNA surveys is already outpacing the rate at which those species can be formally described. Formal description requires a type specimen, a physical preserved example of the species that is deposited in a museum collection and associated with a published morphological description. An eDNA sequence is not a type specimen. It is an indication that something is there. The species it came from must eventually be collected before it can be named. And the number of species indicated by eDNA surveys but not yet collected, not yet seen, not yet described, is growing every year. This is the situation. More than 25,000 new marine species were described between 2000 and 2010, according to the Census of Marine Life. The rate of description has continued at similar pace since then. In each year, the described species added to the catalog of known marine life is a substantial number. In each year, the species indicated by new eDNA surveys, new deep-sea dredging, new ROV footage, and new examination of existing museum collections is a larger number. The ocean is generating new knowledge faster than science can process it, not because science is slow, because the ocean is very large. Consider what it means to know that there are 100,000 seamounts in the ocean and fewer than 500 have been studied. Each seamount is not simply a geological feature, it is an isolated ecological community, potentially harboring species found nowhere else, sitting at the intersection of a particular depth range, a particular water temperature profile, a particular current regime, and a particular geological substrate. The corals that grow on a seamount in the Central Pacific at 1,000 meters depth may be different from the corals on a seamount 200 kilometers away, because the two seamounts have slightly different summit depths, slightly different current exposures, slightly different histories of volcanic activity. The number of ecologically distinct environments in the deep ocean, when you account for all the seamounts and ridges and canyon systems and hydrothermal vent fields and cold seep communities and whale fall communities, is enormous. Each one is a potential source of distinct biodiversity. Almost none of them have been looked at carefully. The Clarion-Clipperton Zone in the Central Pacific is a region of abyssal plain approximately 4,500 kilometers wide and 1,400 kilometers north to south, covering an area roughly twice the size of the continental United States. The floor of this zone is covered in polymetallic nodules, the manganese and iron concretions that accumulate over millions of years, and the fauna living on and among these nodules is distinctive. adapted to an environment that consists of firm substrate scattered across a soft sediment plain. A systematic biological survey of the Clarion-Clipperton Zone, conducted as part of research into the potential environmental impacts of deep-sea mining, found that the majority of animal species collected from any given location in the zone were new to science. In some sampling efforts, the proportion of undescribed species exceeded 90%. In an area of ocean that has been known to exist for decades and that has been the subject of specific survey work motivated by economic interest, 9 out of 10 animals collected were previously unknown. This is the present reality of deep-sea biology. Not a frontier in the sense of something that is being rapidly settled and understood. A frontier in the sense of something whose true extent has only recently become apparent. The deep ocean is not at the edge of being known. It is at the beginning of being approached with tools adequate to the task of learning about it. The learning will take a very long time, and the ocean, in the meantime, will continue to be what it is. The hydrothermal vents will continue to produce their chemical gradients and sustain their communities of tube worms and crabs and archaea. The seamounts will continue to grow their cold water corals over centuries, with or without any observer. The sediment below the seafloor will continue to be inhabited by organisms living in geological time, dividing once every 10,000 years, unbothered by the question of whether anyone knows they are there. The Milky Sea will glow again in the Indian Ocean during the next warm year with the right conditions, covering an area the size of a small country with steady, quiet bioluminescence visible from space, as it has done many times before anyone had a satellite to see it. Something in the bathypelagic will produce a sound that no instrument currently deployed can detect, because the instruments are in other locations, and the sound will propagate through the SOFAR channel and arrive, attenuated and anonymous, at a recording station that will log it as background noise. A snailfish will move across the floor of the Challenger Deep in the dark and cold and pressure, doing what snailfish at that depth do, completely indifferent to the fact that it is among the deepest vertebrates ever recorded and that its physiology is not fully understood. An organism in the sediment somewhere in the South Pacific, 300 meters below the seafloor surface, in sediment that has been compressing for 80 million years, will divide. The process will take perhaps a decade to complete. By the time the cell is divided, the ocean above it will be slightly different from how it is tonight, because everything changes. But the ocean is patient about change the way it is patient about everything. It changes slowly. It holds its inventory with the same indifference it has always had. The mysteries that prove the deep sea is still completely unknown are not mysteries in the sense of puzzles with a hidden solution waiting to be revealed. They are mysteries in the more fundamental sense, descriptions of an environment whose full contents have never been observed, whose full extent has never been visited, whose full chemistry has never been measured, whose full acoustic environment has never been recorded. The ocean is not withholding. It is simply there, in its entirety, at every moment. We have visited a fraction of it. The fraction is enough to tell us, with certainty, that the rest contains things we have not imagined. That is enough to know for tonight. The ocean is not finished. It was not finished before tonight. It will not be finished tomorrow. And what is in it, most of what is in it, is still there, still present, still unnamed, still alive, and very patient. The ocean, before the invention of the sonar, was measured one rope at a time. The word fathom, the unit of depth still used in some nautical contexts, equal to approximately 1.8 meters, comes from an Old English word for the span of outstretched arms, because that was how the rope was measured before it was paid out. Every depth measurement in the ocean's record before 1920 represents a sailor at a rail, holding a marked rope, watching it go down, waiting for it to stop. The Challenger expedition's 492 soundings in 3.5 years represent an enormous investment of time and labor by that measure. What those soundings found, when they were plotted and analyzed, was the first global picture of ocean depth, a world with an average depth of about 3,700 meters, with a maximum at what would eventually be identified as the Challenger Deep in the Mariana Trench, with ocean basins separated by ridges and scarps and seamounts whose existence had been entirely unsuspected. The Challenger scientists named many features. Some of those names persist, but the picture they could build was so coarse, 492 points scattered across an ocean of 361 million square kilometers, that it resembled a sketch made from 492 pixels in a photograph that requires billions of pixels to show its actual content. When echo sounding arrived in the 1920s and became standard equipment on research vessels, the rate of data collection increased dramatically. Ships could now measure depth continuously along their track, producing a profile of the ocean floor rather than isolated points. By the 1950s and 1960s, the accumulated echo-sounding profiles from decades of ship tracks across the global ocean had begun to reveal something extraordinary. The ocean floor was not flat. It had mountains, it had valleys, and running through the center of every ocean basin was a continuous elevated ridge. The discovery of the mid-ocean ridge system is one of the great intellectual achievements of 20th century science, and it came almost entirely from the analysis of acoustic depth measurements. Marie Tharp, a geologist and cartographer at the Lamont Geological Observatory in New York, spent years in the 1950s translating echo-sounding profiles from the North Atlantic into topographic maps of the Atlantic seafloor. She identified in the data a consistent valley running along the crest of the mid-Atlantic ridge, what she called the Rift Valley, and recognized it as evidence that the seafloor at the ridge was being pulled apart rather than compressed. Her colleague Bruce Heezen was initially skeptical. Her maps were disputed. Within a decade, the evidence from other sources had made the truth undeniable. The seafloor was spreading, new crust was being created at the mid-ocean ridges, and the continents were moving apart on the surface of plates of crust that floated on the mantle below. This is the theory of plate tectonics, now fundamental to all of Earth science. It was established primarily by acoustic data from the deep ocean, interpreted by careful analysis of depth profiles, and confirmed by geomagnetic surveys of the oceanic crust. The ocean did not announce that it was the site of the world's largest mountain range, the site of the world's most active geological process, the site of the mechanism by which the surface of the planet is continuously renewed. The data revealed it, slowly, to people who were paying careful attention. The lesson the ocean teaches about itself through the history of plate tectonics is the same lesson it teaches through the history of chemosynthetic vents and the deep biosphere, and the Milky Sea and the bioduct. The ocean holds its secrets not through any active concealment, but through sheer scale and inaccessibility. The secrets are available to whoever looks with the right instruments and interprets the results with sufficient care. But the looking requires enormous investment and patience, and even then the results are often incomplete. A specific example of the incompleteness persisting into the present day: the Puerto Rico Trench, located just north of Puerto Rico in the Atlantic Ocean, is the deepest point in the Atlantic at approximately 8,180 meters. It is also less than 200 kilometers from one of the most intensively studied ocean regions on Earth, the Caribbean and its associated waters, which have been the site of centuries of ship traffic and decades of scientific research. And yet the Puerto Rico Trench, despite its proximity to heavily trafficked waters and its status as the deepest point in the Atlantic, has received fewer direct visits from submersibles or deep-rated ROVs than many locations in the much more distant Pacific. The fauna of the Puerto Rico Trench at its deepest points is essentially unknown. The geology of the trench floor has not been surveyed at meaningful resolution. It is, in the most literal sense, unexplored, despite sitting in what might be called the most studied ocean basin on Earth. The same is true of the South Sandwich Trench in the extreme southern Atlantic, the Romanche Trench along the equatorial Atlantic mid-ocean ridge, and numerous other deep Atlantic features. The Atlantic is often described, relative to the Pacific and Indian Oceans, as a well-studied ocean. Its surface currents, its temperature and salinity structure, its biological productivity, its contribution to the global ocean circulation. These are studied by dozens of research programs operating continuously. Its floor below 3,000 meters is mostly unvisited. The distinction between the surface ocean and the deep ocean, in terms of how well each is known, is one of the sharpest divides in all of science. The surface ocean, the top few hundred meters, the zone accessible to ships, to satellite remote sensing, to buoys and floats and the light of the sun, is studied intensively and understood in substantial detail. The deep ocean, everything below about 1,000 meters, the majority of the ocean by volume, is known primarily through the accumulation of a sparse and geographically uneven record of expeditions, each one visiting a small number of locations for a limited time. Consider the Argo program, which has deployed more than 4,000 autonomous floats across the global ocean, each measuring temperature and salinity as it rises and falls between the surface and 2,000 meters depth. The Argo program is one of the most comprehensive ocean observing systems ever built, and it has transformed the understanding of upper ocean heat content and the thermohaline circulation. It reaches to 2,000 meters. The average ocean depth is 3,688 meters. Below 2,000 meters, below the reach of Argo, the global ocean is essentially unmonitored on a continuous basis, except for a small number of moored instruments and occasional research cruises. A newer generation of deep Argo floats is being developed and deployed, capable of reaching 6,000 meters. This will expand continuous monitoring to the vast majority of the ocean's volume. But as of the mid-2020s, the deployment is in its early stages. The global deep ocean is still, in most of its volume, a place that is visited occasionally and monitored rarely. The question of what happens when someone actually goes somewhere new in the deep ocean, rather than extrapolating from adjacent data, has been answered consistently and dramatically throughout the history of deep-sea exploration. But there is a specific category of answer that has come from deep-sea exploration that is worth tracing in detail, because it concerns the ocean's relationship with human medicine. The deep-sea sponge Halichandria okadai, collected from shallow coastal waters off Japan, produced a compound in 1986 that researchers named halicandrin B. The compound was remarkable for its biological activity. It bound to a component of the cellular machinery that divides chromosomes during cell division, blocking mitosis and killing the cell. This is precisely the kind of mechanism sought in cancer treatment, where stopping the uncontrolled division of malignant cells is the central therapeutic goal. Halichandrin B was too complex and too difficult to synthesize in the quantities required for clinical use. But a simplified synthetic analog, eribulin, was developed and approved by the United States Food and Drug Administration in 2010 for the treatment of metastatic breast cancer. It is used today in chemotherapy clinics in patients whose cancer has stopped responding to other treatments. The organism that produced the lead compound for eribulin, the compound that inspired the chemical series that led eventually to an approved drug, was a sponge. Sponges are among the oldest multicellular animals on Earth, with fossil records extending back more than 500 million years. They are sessile, meaning they cannot move. They cannot run from predators. They cannot fight them off mechanically. Over hundreds of millions of years, in environments where predation pressure is intense, sponges have evolved a chemical defense. They produce compounds that are toxic or repellent to the organisms that would eat them. These compounds are, by selection, bioactive. They interfere with the biology of potential predators in ways that discourage the predators from continuing to eat the sponge. The same interference, in many cases, is medically useful. The deep-sea sponges are of particular pharmacological interest for two reasons. First, they live in environments with intense competition for space on hard substrate. Seamount surfaces, rocky outcrops, hydrothermal vent structures, and where grazing pressure from echinoderms and fish can be significant. The chemical arms race in these environments has produced an extraordinary diversity of bioactive compounds. Second, the deep-sea environment is so physically stable, constant temperature, constant salinity, constant pressure, that the organisms living there have had millions of years to refine their chemistry without the additional challenge of adapting to environmental variability. The result is chemical sophistication of an unusual degree. The discodermalide story illustrates the potential and the difficulty simultaneously. The compound was first isolated in 1990 from a deep-sea sponge of the genus Discodermia, collected at about 30 meters depth in the Bahamas. Not deep ocean by the standards of this documentary, but a marine sponge nonetheless. Discodermalide showed potent anti-tumor activity in laboratory testing, outperforming several existing chemotherapy drugs in cell culture experiments. Clinical development was pursued by a pharmaceutical company, and early clinical trials showed activity but also significant toxicity. The compound did not progress to approval, but it contributed substantially to the understanding of the tubulin binding mechanism it targets, and analogs derived from discodermalide continue to be studied. The fraction of marine organisms that have been systematically screened for bioactive compounds is very small. Most of what has been screened comes from shallow coastal environments and from the limited samples collected during deep-sea research expeditions. The deep-sea organisms specifically, the organisms of hydrothermal vent communities, of cold seep communities, of hadal trenches, of abyssal plain sediments, are among the least screened. An organism like the alvanellid worms that live on the surface of hydrothermal vent chimneys, surviving temperatures that would kill most animals within seconds, must have evolved biochemical machinery of unusual robustness to maintain cellular function at those temperatures. That machinery, the heat-stable enzymes, the membrane lipid compositions, the molecular chaperones that prevent protein denaturation, represents potential biotechnological and pharmaceutical material that has barely been examined. The Taq polymerase enzyme, isolated from a thermophilic bacterium, Thermus aquaticus, living in hot springs at Yellowstone National Park, made the polymerase chain reaction possible. PCR, the method by which tiny amounts of DNA can be amplified to quantities sufficient for analysis, is the foundation of modern molecular biology, forensic science, and clinical diagnostics. It is used billions of times per year in laboratories worldwide. Its central enabling enzyme came from a heat-adapted organism. The deep-sea hydrothermal vent communities harbor organisms adapted to temperatures far exceeding those at Yellowstone, in a chemical environment far more complex. What their biochemistry contains, in terms of catalytically robust and chemically sophisticated molecules, is almost entirely unknown. The argument here is not that the deep ocean is a pharmacy waiting to be opened. The argument is more specific and more well-grounded. Every time a deep-sea organism has been collected and its chemistry examined carefully, the chemistry has turned out to be interesting in ways that have implications for medicine, biotechnology, or fundamental biochemistry. The rate of yield from deep-sea organism chemical screening is high enough to justify the effort of the screening, and the fraction that has been screened is small enough that the total amount remaining is genuinely large. We are, in the pharmacological sense as in every other sense, at the beginning of a survey rather than near the end of one. The ocean has not revealed its chemical contents. It has shown us samples. The samples have been good enough to establish that the rest is worth looking at, and then we have mostly not looked. The deep-sea Greenland shark occupies a different position in the catalog of deep-sea mysteries. It is not unknown in the sense of being newly discovered. The species, Somniosus microcephalus, has been documented for centuries in the North Atlantic and Arctic oceans, caught occasionally in fishing nets and observed by researchers. But its biology, until recently, was profoundly mysterious in a specific way. No one knew how long it lived. The Greenland shark moves slowly, grows slowly, and reproduces slowly. These are characteristics associated with long-lived species, and researchers had long suspected that the Greenland shark was exceptionally long-lived by the standards of vertebrate animals. But determining the age of a shark requires examining structures that accumulate growth layers over time, the way tree rings accumulate in wood. Sharks do not have the calcified vertebrae that show clear annual growth rings in bony fish. Determining the age of a Greenland shark from its anatomy was, for most of the history of shark biology, not possible. In 2016, a team of Danish researchers published a study using a different method. Radiocarbon dating of the proteins in the eyelens of Greenland sharks, combined with knowledge of how radiocarbon levels in the atmosphere changed after the nuclear weapons tests of the 1950s and 1960s. The nuclear tests produced a spike in atmospheric radiocarbon that was incorporated into the eyelens protein of animals alive during that period. By looking for the presence or absence of that spike in the lens proteins of sharks of different sizes, and combining this with known growth rates, the researchers could estimate the ages of the sharks they analyzed. The results were extraordinary. The largest female Greenland shark in their sample, approximately 4 meters in length, was estimated to be between 272 and 512 years old, with a central estimate of about 400 years. The median estimated age of the mature females in the sample was approximately 150 years. The Greenland shark is the longest-lived vertebrate animal known, living potentially longer than any mammal, any bird, any reptile, any bony fish. An individual alive today may have been born before the European settlement of North America, before Isaac Newton formulated the laws of motion, before the Baroque period in European music. The Greenland shark's longevity was not suspected. It was not predicted. It was revealed by the specific application of a specific analytical technique to a specific sample of preserved eye lenses from a species that had been known to science for a very long time. The animal was familiar. Its fundamental biology was not. This is the consistent pattern. The familiar animal, examined with new techniques or in new ways, turns out to be far stranger than the record indicated. The ocean is full of animals that are known in the sense of having a scientific name and a basic morphological description, and unknown in the sense that their physiology, behavior, ecology, and life history are either unstudied or studied only in the most basic outline. The Census of Marine Life, a large international research program that operated from 2000 to 2010 and involved more than 2,700 scientists from 80 countries, set out to answer three fundamental questions about the ocean's biology: what lived there, what lives there now, and what will live there in the future. The first question addressed historical records and museum collections. The second required new sampling and analysis. The third was a modeling question dependent on the answers to the first two. What the census found, when it concluded in 2010, was published in a series of papers and summaries that painted a clearer picture of marine biodiversity than had previously existed. The total number of known marine species at the conclusion of the census was approximately 250,000. The estimate of total marine species, including those not yet described, ranged from 700,000 to more than 1 million. The majority of the unknown species, the census concluded, are in the deep sea. The reasoning behind this conclusion is straightforward. The shallow coastal zones of the ocean, the coral reefs, the kelp forests, the shallow continental shelves, have been studied intensively for decades and in some regions for centuries. These environments are relatively accessible to divers and ships, and the organisms in them are visible in ordinary light. The proportion of known to unknown species in shallow coastal environments, while still substantial, new coral reef species are described every year, is significantly higher than in the deep sea. In the deep sea, where every sample requires expensive equipment, long ship voyages, and specialized expertise, the proportion of unknown to known is reversed. For every deep sea species described, there are many more waiting to be found. The deep sea portion of the census drew on new sampling efforts from multiple regions and depth zones. In the Pacific, expeditions sampled abyssal plains, seamounts, and hadal trenches. In the Atlantic, deep coral reefs and continental slopes were surveyed. In the Arctic, sub-ice communities were sampled for the first time using ice-capable research vessels. In every region surveyed, significant numbers of species new to science were collected alongside the known fauna. A census of the deep sea floor undertaken during the census program found that in the Pacific abyssal plain, a standardized sampling effort collected organisms from multiple phyla at each site, with a significant proportion of the species collected being undescribed. The sampling, standardized across sites, allowed comparison of diversity at different locations and depths. What emerged was a picture of deep-sea diversity that was higher than many researchers had expected, and that showed consistent patterns of zonation, different communities at different depths, at different distances from the ridge system, at different positions relative to productivity zones in the surface ocean above. The diversity of the deep sea is not understood. This is a precise scientific statement. The number of species, their distributions, their ecological relationships, their evolutionary histories, all of these are estimated with wide uncertainty, inferred from sparse sampling, extrapolated from limited data. The species diversity of a hectare of tropical rainforest can be estimated with some confidence from intensive surveys. The species diversity of a hectare of abyssal plain cannot be estimated with anywhere near the same confidence, because the sampling density required to characterize deep-sea diversity adequately has never been achieved at any location. There is a specific biological phenomenon in the deep sea that has attracted attention precisely because it is unexpected and unexplained: the pattern of diversity at the greatest depths. Classical ecological theory and the evidence from shallow marine environments predicts that species diversity decreases in more extreme environments. The logic is intuitive. Extreme conditions, very cold, very hot, very saline, very dry, reduce the number of species capable of surviving and therefore reduce diversity. The deep sea is extreme by several measures: cold, high pressure, dark, food limited. The prediction would be low diversity. But the deep sea sediment fauna does not consistently follow this prediction. At intermediate depths, roughly 1,000 to 3,000 meters, diversity in some taxa, particularly in small crustaceans called isopods and in polychaete worms, is very high. The diversity then drops off at the greatest depths, but the high diversity at intermediate depths is itself surprising given the conditions, and the mechanisms that maintain it are debated. Some researchers attribute it to the high spatial heterogeneity of the deep sea floor. The variety of different microhabitats created by bottom currents, sediment composition, and local biological activity. Others attribute it to the stability of the deep-sea environment over evolutionary time, which has allowed the accumulation of species without the periodic extinctions that climate variability causes in shallower environments. The question is not settled. The deep-sea polychaete worms, a particularly diverse group of segmented worms that inhabit soft sediments, have been estimated, based on extrapolation from sampling data, to include potentially tens of thousands of species globally, of which only a few thousand are described. This estimate carries wide uncertainty. It is based on species-area relationships derived from shallow coastal sampling, extrapolated to the much larger area of the deep ocean. The extrapolation may or may not be valid, but the deep-sea polychaete diversity is almost certainly large, and the known diversity is almost certainly a small fraction of the total. The deep sea is also the oldest habitat on Earth in the sense of being the most continuously inhabited. While coastal environments have been repeatedly disrupted by sea level changes, glaciation, and tectonic events that flooded and drained shallow seas, the deep ocean has maintained broadly similar conditions for tens to hundreds of millions of years. This stability has allowed the accumulation of extremely ancient evolutionary lineages, groups of organisms that diverged from their closest relatives at very early points in the history of multicellular life, and have persisted since in the deep ocean as relics of evolutionary history. The nautilus, often discussed as a living fossil because its shell morphology resembles that of ammonites extinct for 65 million years, lives in the deep slope environments of the Indo-Pacific, at depths of 100 to 700 meters. The horseshoe crab, more accurately described as a living relic of the Chilicerata lineage rather than a crab, occupies coastal environments, but has relatives in deep-sea habitats. The coelacanth, the famous living fossil fish of the Indian Ocean, lives at depths of 150 to 700 meters, along submarine cliffs and deep rocky slopes. These animals represent lineages that have survived for hundreds of millions of years in environments where the ocean's characteristics have been sufficiently stable to maintain their niche. The deep ocean is not only large and dark and cold, it is also old. Its biological communities are old. The organisms in it have been refining their adaptations for longer, in most cases, than any shallow water community. The evolutionary inventory of the deep ocean, the sum total of the adaptations, behaviors, biochemistries, and ecological relationships that have developed there over the history of life on Earth, is one of the richest stores of biological information that exists, and it is mostly unread. One of the clearest demonstrations of this richness came from an unexpected source, the hydrothermal vent shrimp of the Mid-Atlantic Ridge. When hydrothermal vents were first discovered at the East Pacific Rise in 1977, the vent communities of the Atlantic were not yet known. When Atlantic vents were subsequently discovered at the Mid-Atlantic Ridge in the 1980s, the biological communities were found to be different from those of the Pacific, reflecting the different evolutionary histories of the Atlantic and Pacific Ocean basins. Among the organisms at the Atlantic vents was a shrimp, Rimicaris exoculata, that was found in swarms of thousands of individuals around the vent chimneys. Rimicaris exoculata has no compound eyes. The eye structure visible on its back is not a conventional image-forming eye at all, but a highly modified eye with an enormously enlarged retina adapted to detect extremely faint light, possibly the faint glow of chemiluminescence from the vent fluids themselves, or possibly some other very low-intensity light source associated with the vent. The shrimp uses this unusual visual system to navigate around the vent in an environment where the temperature gradient between lethal and survivable is only a few centimeters, and where staying too close to the chimney means death and staying too far means starving. The modified eye appears to allow the shrimp to detect thermal radiation from the vent, light produced by the very high temperatures of the vent fluid, and use it to maintain its position in the survivable thermal zone. The evolutionary path from a normal compound eye to a photoreceptive organ capable of detecting thermal infrared radiation from a hydrothermal vent, in a lineage that probably colonized vent environments after the evolution of the deep-sea shrimp lineage, represents an adaptation so specific and so remarkable that it was not predicted and would not have been imagined before the shrimp was collected and its anatomy examined. The deep ocean produces these adaptations. It produces them constantly, in organisms living in environments whose specific properties have shaped them over millions of years. The adaptations, discovered one by one as the organisms are collected and examined, are consistently surprising. The ocean teaches patience by example. It has been doing this for longer than anything else on Earth. Every organism in it has been shaped by time in a way that nothing above the surface has been, because the surface changes faster and the deep does not. The adaptations are precise. They are refined. They are solutions to problems that humans have not always recognized as problems, in environments that humans are still learning to describe. And the organisms carrying those adaptations are, in most cases, still there, still alive, still doing what they do, still waiting to be found and studied and understood, in whatever patient way the deep ocean holds everything that belongs to it. There is a specific kind of deep-sea mystery that has to do not with what we have not yet found, but with what we have found and cannot explain. The deep-sea acoustic record contains these mysteries, as already described. The biological record contains them as well, in the form of organisms whose behavior in the wild has been observed but remains unexplained. The giant squid, Architeuthis dux, is among the most famous of the deep-sea animals whose life history remains largely mysterious, despite being known to science for more than a century. Giant squid can reach lengths of 13 meters from the tip of the mantle to the end of the longest tentacles, making them among the largest invertebrates ever documented. They are known from stranded specimens, from body parts found in the stomachs of sperm whales, and, since 2004, from a small number of photographic and video observations from submersibles and baited camera systems in the deep Pacific. What is not known about giant squid is significant. Their diet in the wild is inferred from remains found in sperm whale stomachs and from the few observations of live animals, which suggest they are active predators feeding on fish and smaller squid, but the specific composition and frequency of their feeding is unknown. Their reproductive behavior has never been observed. Females carry fertilized eggs internally, but where and how they spawn, what the juveniles look like and where they live, and how young squid grow from the small juvenile stage to the enormous adult stage, none of this has been observed. Their population size is genuinely unknown. The number of giant squid in the world's oceans has been estimated from the sperm whale stomach data, but with enormous uncertainty. They could be extremely rare or moderately common. The evidence does not resolve this. What is known is that sperm whales, which dive to depths of more than 2,000 meters and can hold their breath for up to 90 minutes, pursue giant squid actively and depend on them as a major component of their diet. The evidence for this is the beaks of giant squid found in sperm whale stomachs. Squid beaks are indigestible and accumulate in the stomach over years of feeding. A single sperm whale stomach can contain dozens of giant squid beaks. The population of giant squid in the ocean must be large enough to sustain the predation pressure from hundreds of thousands of sperm whales, each consuming a diet substantially composed of large squid. Whatever the population of giant squid is, it is not small. Yet giant squid are almost never seen alive. The 2004 photographs, taken by Japanese researchers Tsunemi Kubodera and Kyoichi Mori, using a baited camera system deployed at 900 meters depth in the Pacific, were the first photographs ever taken of a living giant squid in its natural habitat. Before 2004, all knowledge of the living animal came from stranded individuals, from animals accidentally caught in fishing nets, from descriptions of animals that attacked or were observed near ships, and from inferences based on sperm whale stomach contents. After 2004, a handful of additional observations have been made, including a video taken by the submersible used during a NOAA expedition to the Gulf of Mexico in 2019. But the total number of observations of living giant squid in their natural habitat remains in the single digits. For an animal that must exist in populations sufficient to feed hundreds of thousands of large predators, it is almost invisible to us. The invisibility is not an artifact of the animal's small population. It is an artifact of the scale of the environment in which it lives and the difficulty of being in the right place at the right time with the right equipment. The giant squid lives in the mesopelagic and bathypelagic zones of the open ocean, in a volume of water that is vast beyond any intuitive sense of scale. Deploying a camera at the right depth at the right location to intercept one of these animals as it passes requires either extraordinary luck or extraordinary coverage. And the coverage we have of the deep ocean's volume with cameras and sensors is so sparse that extraordinary luck is the mechanism most of the observations have relied on. The mesopelagic zone alone, the layer between roughly 200 and 1,000 meters depth, has a total volume of approximately 355 million cubic kilometers. The collective volume of all the submersibles and ROVs that have ever operated in that zone, all of their cameras, all of their recorded footage, represents a vanishingly small sample of the available space. Finding a giant squid in that volume without a specific tracking method is roughly equivalent to finding a specific individual in the population of Earth by walking around at random. The giant squid is known and unknown simultaneously. Known as a species, known in morphology, known in its existence as a major component of sperm whale diet. Unknown in its behavior, its reproduction, its development, its population dynamics, its ecology in the wild. The known and the unknown sit beside each other in the scientific literature, and the gap between them is the ocean. The Magnapinna squid presents a different kind of mystery. The species, or species group, the taxonomy is not fully resolved, was first identified from specimens caught in trawls in the 1990s. It belongs to a family of squid called Magnapinnidae, characterized by very large fins that extend along almost the entire length of the mantle, giving the squid an unusual appearance, and by extremely long arms and tentacles with elbow-like bends in the middle. In live observations from ROVs, Magnapinna squid have been seen drifting in the water column with their arms and tentacles hanging downward from the elbow bends, in a posture not seen in any other squid. The purpose of this posture is unknown. The total body of observations of living Magnapinna is small. Fewer than 20 ROV encounters have been published, and the diet, distribution, population, behavior, and ecology of the animal are essentially unknown. What is known is that Magnapinna has been recorded at depths ranging from about 1,000 meters to nearly 5,000 meters, suggesting it occupies the full vertical extent of the bathypelagic zone. It has been observed in the Pacific, Atlantic, and Indian Oceans, suggesting a global distribution. Its very large fins and the elbow posture of its arms suggest a specific ecological strategy. But what that strategy is? Passive ambush predation? Current-based drift feeding? Something entirely different. Is not established. The animal drifts in the dark and does what it does, and we do not know what that is. The ocean does not reserve its mysteries for the largest or most dramatic animals. The microscopic organisms of the deep ocean present mysteries of comparable depth and greater breadth. The archaea of the deep ocean, single-celled organisms in the domain distinct from both bacteria and eukaryotes, discovered by Carl Woese and colleagues in the 1970s through RNA sequence analysis, are now known to be extraordinarily abundant throughout the water column and sediment of the deep ocean. Studies using molecular sequencing methods have shown that archaea can make up as much as 40% of the microbial cells in deep-sea water samples, with abundances of 100,000 to 1,000,000 cells per milliliter of seawater. These archaea, particularly a group called Thaumarchaeota, now sometimes classified as Nitrososphaeria, are known to oxidize ammonia in the deep ocean, converting it to nitrite as part of the global nitrogen cycle. This process, called nitrification, affects the availability of nitrogen in a form usable by phytoplankton and therefore influences the productivity of the surface ocean. The archaea carrying out this process are, in terms of sheer abundance and geochemical impact, among the most important organisms in the ocean. Their discovery in the early 2000s as a dominant component of the deep ocean's microbial community was a major revision of the understanding of ocean biogeochemistry. But the metabolism of the deep-sea archaea is not limited to nitrification. Molecular surveys have identified genes in deep-sea archaeal communities suggesting metabolic capabilities for which no cultured representative exists. Metabolic pathways inferred from DNA sequences but not yet demonstrated in any organism that has been grown in a laboratory. What these pathways do, what conditions activate them, what compounds they produce and consume, these questions cannot be answered without growing the organism, which for most deep-sea archaea has not been achieved. The culture of deep-sea microorganisms is technically challenging because the organisms are adapted to conditions of high pressure, cold temperature, and very low nutrient concentrations that are extremely difficult to replicate in a laboratory setting. The gap between what the DNA sequences of deep-sea microorganisms suggest they can do and what has been demonstrated in laboratory culture is one of the largest unexplored territories in contemporary microbiology. The sequences are available. Environmental DNA sequencing has produced enormous catalogs of microbial genetic diversity from the deep ocean. The organisms in most cases are not. The gap is not expected to close quickly. This is the specific character of the unknown in the deep ocean. It is not the unknown of science fiction, where the mystery is presented as terrifying or threatening or deliberately concealed. It is the unknown of genuine documented incompleteness. The record shows that something is there. The record cannot show what it does or how it lives, and the means to answer the question are available in principle, but have not yet been deployed at the necessary scale. The deep ocean is unknown in the way that any large archive of undocumented material is unknown, not because it is hiding, but because no one has yet had the time and resources to read it all. The ocean's deepest residents, the organisms of the Hadal Zone, the trenches that reach below 6,000 meters, represent a specific frontier of biology that is actively being explored and that is producing consistent surprises. The Hadal Zone is defined as the depth range below 6,000 meters, which includes only the deepest parts of the world's ocean trenches. It covers a small fraction of the ocean's total area, roughly 0.2% of the global seafloor, but it occupies a unique position in the ocean's ecological structure because the environmental conditions there, in terms of pressure, food availability, and isolation, are more extreme than those at any other depth. The pressure at the bottom of the Challenger Deep is approximately 1/100 atmospheres, 1/100 times the pressure at sea level. This pressure would crush any conventional container not specifically designed to withstand it. It compresses water measurably, altering its density and the speed of sound that travels through it. It compresses the lipids and proteins in cell membranes, altering their fluidity and their function. The organisms that live at Hadal depths have cell membranes and enzyme structures specifically adapted to maintain function at pressures that would denature the equivalent structures in any shallow water organism. The biochemistry of this adaptation is studied through the analysis of collected specimens, but the full molecular picture remains incomplete. The discovery that snailfish of the family Liparidae are the deepest fish in the ocean, reaching depths of nearly 8,400 meters in the Mariana Trench and apparently being the dominant fish predator in Hadal communities, was established through targeted expeditions with baited cameras and traps. The snailfish at these depths are pale, translucent, and gelatinous in appearance. Their anatomy is adapted to minimize the cost of maintaining a rigid body structure under extreme pressure. They are surprisingly active, moving efficiently across the Hadal sediment in pursuit of the amphipod crustaceans that aggregate around food falls at these depths. One of the most striking findings from Hadal biology is the chemical contamination. In 2017, a study published in the journal Nature Ecology and Evolution by Jameson and colleagues analyzed the tissue of amphipods collected from the Mariana Trench and the Kermadec Trench. The amphipods, living at depths of more than 10,000 meters in some of the most remote environments on Earth, contained concentrations of polychlorinated biphenyls, PCBs, and polybrominated diphenyl ethers, PBDEs, that were dramatically higher than the concentrations found in amphipods from the nearby open ocean, and in some cases higher than concentrations measured in heavily polluted coastal industrial zones. The explanation is not that the trenches are receiving direct pollution input. The trenches are far from any pollution source. The explanation is that PCBs and PBDEs, produced in large quantities by industrial activity primarily in the mid-20th century, are highly resistant to biological degradation and are sequestered in organic matter that sinks through the water column. The marine snow that carries organic material to depth carries these persistent organic pollutants with it, and the trench, as the lowest point in the basin, receives the concentrated accumulation of decades of sinking particles. The amphipods eat whatever organic material arrives at the trench floor, and they accumulate the pollutants in their fatty tissue. The deep trench, isolated by its depth and geography, has become a reservoir for compounds produced at the surface and carried down by the biological pump. The deepest and most remote environment on the planet contains traces of a specific industrial era in human history, preserved in the tissue of animals adapted to extreme depth. This is not a metaphor. It is a measured chemical result from the analysis of real tissue from real animals collected at real depth. The Clarion-Clipperton Zone presents a different kind of challenge to the unknown. This vast abyssal plain in the Central Pacific, approximately the size of the continental United States, is at the center of international plans for deep-sea mining of the polymetallic nodules that cover its floor. The nodules contain manganese, nickel, cobalt, and copper in concentrations that make them economically attractive. The technology to harvest them at commercial scale is being developed. The environmental consequences of harvesting them are not yet fully understood. A survey of the Clarion-Clipperton Zone biology, conducted in the 2010s as part of the environmental baseline work required by the International Seabed Authority before any mining licenses can be granted, produced data that clarified the situation in a way that was not expected to be as striking as it turned out to be. The survey collected animals from multiple sites across the zone using sediment cores and epibenthic sleds. The taxonomists who analyzed the samples found that the majority of the species collected were new to science. At some sites, the proportion of undescribed species exceeded 90%. This means that the environmental impact assessment for deep-sea mining in the Clarion-Clipperton Zone must be conducted without baseline knowledge of the majority of species that live there. The impact statement for a mining operation is supposed to establish what exists before mining, document what is damaged or destroyed, and assess whether the damage is acceptable. If the species that live in the zone cannot be identified because they have no names and no descriptions, then the impact statement cannot fully document what is at risk. The environmental review process, which depends on existing scientific knowledge, encounters a fundamental gap in that knowledge precisely where the gap matters most. This is the practical consequence of the deep ocean being unknown. It is not only a matter of scientific incompleteness, it is a matter of decision-making in the absence of information, in circumstances where the decisions are irreversible, because the removal of the nodules from the Clarion-Clipperton Zone, if carried out, will alter the seafloor environment permanently. The nodules grow at a rate of roughly 1 centimeter per million years. A nodule field cleared by industrial mining will not recover on any human timescale. The deep ocean is unknown in a way that has consequences that are arriving now, not at some indefinite future point when the ocean might finally be fully surveyed. The consequences are arriving because human activity, mining, bottom trawling, climate change, pollution, is affecting the deep ocean in ways that cannot be adequately assessed because the baseline is not established. And the baseline will not be established before some of the activity has already proceeded. This is not an argument for paralysis. It is an argument for pace, for speeding up the rate of basic deep-sea biological survey to bring the baseline knowledge closer to adequate before decisions with permanent consequences must be made. The rate of deep-sea survey is increasing, driven by new technology and increasing international concern about the management of deep-sea resources. But the gap between what is known and what would be needed to make fully informed decisions remains very large. The ocean does not advocate for itself. It contains what it contains. It changes in response to what is added to it or taken from it. It does not signal when something important is lost. That is part of what makes it worth knowing. The history of how individual creatures have revealed themselves to science, each in its own way and on its own schedule, gives a particular texture to the broader picture of what remains unknown. The coelacanth, Latimeria chalumnae, is the most famous case. The coelacanths were known from their extraordinarily detailed fossil record as a group of lobe-finned fish abundant in the Devonian and Cretaceous periods, among the most recognizable animals in the fossil record of any marine fish group. They disappeared from the fossil record approximately 65 million years ago, coincident with the end Cretaceous mass extinction event. Paleontologists considered them extinct. The fossil record gave no indication otherwise. In December 1938, Marjorie Courtenay Latimer, the curator of a small museum in East London, South Africa, was examining a catch brought in by a fishing trawler when she found a fish she did not recognize. It was large, about 1.5 meters long, and heavily built with thick, tough scales, a distinctive pattern of spots, and an unusual arrangement of its fins. She sketched it and sent the sketch to J.L.B. Smith, a chemistry professor and amateur ichthyologist at Rhodes University. Smith recognized the animal immediately from the sketch. It was a coelacanth. A fish that had been extinct for 65 million years was alive and had just been caught in a fishing net off the coast of South Africa. Smith spent years searching for a second specimen. The discovery had been made near the mouth of the Chalumna River, but the type of habitat most likely to support the fish was unclear from the single specimen. In 1952, a second coelacanth was found near the Comoro Islands northwest of Madagascar, and it became apparent that the species had a significant population in the deep waters of the western Indian Ocean, probably along submarine cliffs and rocky slopes at depths of 150 to 700 meters. Subsequent research established that the Comoro Island population was the main known population of Latimeria chalumnae. In 1997, a second species, Latimeria monadoensis, was discovered in the waters off Sulawesi, Indonesia, essentially by chance. An ichthyologist on honeymoon noticed a coelacanth in a fish market and recognized it. Two living species of a group thought extinct for 65 million years. The coelacanth is not a rare example of a false extinction determination. It is the most famous example of a general principle. Organisms assumed to be extinct because they are absent from the fossil record, since a particular date may simply have moved to environments where they were not being observed and where their remains were not preserved in sediments likely to be sampled by paleontologists. The deep ocean, in particular, is an environment from which the fossil record is sparse and discontinuous. The organisms of the deep ocean can disappear from the fossil record without disappearing from the ocean, if the conditions for their preservation change. The Indonesian coelacanth population is genetically distinct from the Comoros population, having been separated long enough by oceanic geography to diverge substantially. It was found in 1997. Before 1997, it was not known to exist. After its discovery, researchers recognized that the deep, rocky slopes of the Indo-Pacific basin, if systematically surveyed for coelacanths, might well contain additional populations at other locations. Surveys have since documented individual coelacanths in South Africa, in Tanzania, in Mozambique, and in Kenya, populations or individuals not previously known, suggesting that the total range of the species and its population is larger than the Comoros discovery initially implied. The extent of the coelacanth's range is still being determined. The megamouth shark, Megachasma pelagios, was discovered in 1976 when one became entangled in a sea anchor deployed by a United States Navy vessel off the coast of Hawaii. The shark was enormous, nearly 4.5 meters long, and entirely unlike any known shark species. Its mouth was extraordinary, disproportionately large with a silver-lined interior and hundreds of small teeth adapted for filter feeding. A shark species capable of filter feeding at the scale of a whale shark, previously entirely unknown, was discovered when one accidentally entangled itself in naval equipment. Before 1976, the megamouth did not exist in the scientific record. After 1976, analysis of its anatomy suggested it was a member of an isolated lineage with no close living relatives, having diverged from other shark groups tens of millions of years ago. Since 1976, megamouth sharks have been found stranded on beaches, caught incidentally in fishing nets, and observed briefly by ROVs, primarily in the Pacific and Indian Oceans. As of the mid-2020s, fewer than 200 specimens have ever been recorded. The shark lives in the mesopelagic zone and makes daily vertical migrations, ascending toward the surface at night to feed on concentrations of plankton and descending to daytime depths of 150 to 200 meters. Its population is unknown. Its reproductive behavior has never been observed. Its full geographic range is imprecisely known. A shark nearly 5 meters long, filter feeding in the mesopelagic zone of tropical and subtropical oceans worldwide, was entirely outside the scientific record until 1976 and remains very poorly known in 2024. This is not an accident of insufficient effort. It is a consequence of the inaccessibility of its habitat and the infrequency with which the animal comes close enough to the surface to be accidentally encountered. The megamouth lives in a zone of the ocean that is routinely transited by ships without any instrumentation capable of detecting it. It moves through the water column on a daily cycle. It is apparently present in multiple ocean basins. It has been seen, in its entire history of scientific documentation, fewer than 200 times. The goblin shark, Mitsukarina ostoni, is another example of an animal known from the fossil record and then found alive. The genus was known from fossil teeth dating to the Cretaceous. A living specimen was discovered in 1898 off the coast of Japan, caught in deep water by a fisherman who brought it to a scientist. The living goblin shark has a remarkable anatomy, a flattened, elongated rostrum projecting forward from the head, the snout from which the animal takes its common name, and a highly protrusible jaw that can extend rapidly forward to capture prey. The jaw mechanics were not understood from preserved specimens because the extension mechanism requires muscle tissue that is lost in preservation. The full extent of jaw protrusion in living animals was only documented in the 21st century from footage of living or freshly dead specimens. Each of these discovery stories, the coelacanth, the megamouth, the goblin shark, the barreleye, the vampiroteuthis, the rimicaris shrimp, follows the same arc. The animal existed before it was known to science. It was going about its existence in the deep ocean, or the mesopelagic, or the deep slope, doing what it does. It became known to science when an encounter occurred, usually accidental, with a human or human instrument in the animal's environment. After the encounter, the animal was studied with whatever methods were available, And the picture that emerged was consistently more complex and more surprising than anything that had been anticipated. The pattern does not stop. The rate of new deep-sea species descriptions has not decreased. The rate of new ecological discoveries, new community types, new behaviors, new associations, new chemical functions has not decreased. The surprises are arriving as fast as the observations are being made, which is a mathematical way of saying that the rate of discovery is limited not by the availability of things to discover, but by the rate at which the observations are being made. This is a specific claim, and it has a specific implication. The ocean is not close to being fully known. It is close to having a catalog of its major categories, the major ecosystem types, the major faunal groups, the major geological features. Within those categories, the specific content, the species, the behaviors, the biochemistries, the ecological relationships, is largely unknown, and the categories themselves are almost certainly incomplete. The history of deep-sea biology is a history of new categories being added when observations are made that do not fit the existing framework. What was known in 1977, before the hydrothermal vents, was wrong about the energy basis of deep-sea ecosystems. What was known in 1993, before the full extent of the deep biosphere was established, was wrong about the habitability of the sub-sea floor. What was known in 1999, before the lost city vents were found, was wrong about the diversity of hydrothermal systems. What was known about Greenland shark longevity before 2016 was wrong. What was known about barreli anatomy before 2008 was wrong. What was known about the bioduct's source before 2014 was wrong. The ocean keeps correcting the record. It does so on its own schedule, revealing what it contains when the conditions, the right instrument at the right place at the right time, are finally met. The corrected record is always larger and stranger than the previous one, and every correction implies that the next correction, the next surprise, the next category-defining discovery is out there in the ocean somewhere, waiting for the moment when someone arrives in the right place with the right instrument. The ocean is patient. It has been patient far longer than any of the institutions studying it have existed. It was patient before the first ship was built. It was patient before the first human looked at it from ashore and wondered what was underneath. And it is patient still, in the dark and the cold and the pressure of its deepest places, holding what it holds, waiting with no sense of urgency for the next person who comes to look. The way the deep ocean relates to time is one of its most unusual characteristics, and it is relevant to why the deep ocean remains so unknown. The relevant timescales of the deep ocean do not match the timescales of human attention and human funding cycles. A cold water coral colony on a seamount at 800 meters depth may be 2,000 years old. It began growing approximately at the time of the Roman Empire. It has grown continuously since, extending its branches by a few millimeters each year, persisting through medieval warming periods and the Little Ice Age and the Industrial Revolution and 2 world wars, indifferent to all of it. A survey of that seamount conducted today captures one moment in the coral's life. A repeat survey 10 years later captures another. The ecological dynamics of the coral community, how the colony grows, what colonizes it, how it changes in response to deep water temperature shifts, operate on timescales of centuries to millennia. No research program has the budget or the continuity to track them in real time. The deep-sea sediment record is different in its timescale. Each centimeter of sediment represents thousands of years of accumulation. The organisms in the sediment, the chemical signatures in the carbonate shells of foraminifera, the layer of soot from major volcanic eruptions, the trace metals deposited during warm and cold oceanic periods, all of this is a continuous record of Earth's history stored in the seafloor. Scientists read this record by drilling cores and analyzing the material layer by layer, each layer corresponding to a specific period in Earth's past. What they recover from those cores, the ancient ocean chemistry, the ancient temperature, the ancient biological assemblages, is information that no other archive preserves. The ocean floor is Earth's memory. It stores the conditions of past oceans, past climates, past biospheres, in a form that can be analyzed and dated. The ice cores from Greenland and Antarctica preserve about 800,000 years of atmospheric history. The deep-sea sediment record preserves tens of millions of years. For the history of the deep ocean itself, the changes in its circulation, its chemistry, its biological communities, the sediment record is the only record. And the sediment record has been sampled at only a fraction of the locations where it exists, with only a fraction of the methods now available to analyze it. The ancient sediment contains microorganisms that were alive millions of years ago and left their remains, their cell walls, their shells, their organic molecules, in the record. Some of what they left behind can be read as a chemical signature of the conditions in which they lived. The temperature of the water, its chemistry, its nutrient status. Some of what they left behind is still not fully interpretable. Molecules whose biological origin is understood, but whose paleoceanographic meaning remains unclear. The sediment archive is being read layer by layer at an increasing number of locations, but the reading is slow relative to the depth of the archive. What the sediment cores reveal across every ocean basin is that the ocean has been different in the past, substantially different. The deep ocean of the Cretaceous period, the era of the dinosaurs, was warmer, lower in oxygen, differently stratified than the modern ocean. The chemistry of its seawater was different. Its biological communities were different. The transition from the warm, oxygen-depleted Cretaceous ocean to the cold, oxygenated modern ocean was a process that took tens of millions of years and involved the evolution of new biological communities adapted to the new conditions. The deep ocean that exists today, the one with its snailfish and its tube worms and its Vampiroteuthis, is a relatively recent ecological configuration by the standards of Earth history. And it is itself changing in response to the chemical and physical changes occurring at the surface. The aragonite saturation horizon, the depth below which calcium carbonate, the material from which most marine shells are made, begins to dissolve rather than precipitate, is shoaling in the ocean. It is moving toward shallower depths in response to the absorption of anthropogenic carbon dioxide, which makes seawater more acidic and reduces the saturation state of carbonate minerals. Coldwater corals, which build their skeletons from aragonite, are living at depths that will become undersaturated for aragonite within decades in some ocean regions. The corals do not know this. They are building their skeletons as they always have. The chemical environment they depend on is shifting beneath them. The consequences for deep-sea coral communities are not fully assessed. Some species may have physiological mechanisms that partially compensate for reduced carbonate saturation. Some will not. The coldwater coral reefs, biological structures thousands of years old, providing habitat for hundreds of associated species, may begin to dissolve in the coming centuries as the chemistry of the water surrounding them changes. This is not a dramatic event with a visible beginning. It is a slow process whose effects will be visible across timescales longer than any individual research program. There is a particular kind of loneliness to the prospect. A coral that has been growing in the dark for 2,000 years, in water whose chemistry has been stable throughout that period, will begin to experience the effects of a change that originated at the ocean's surface and propagated downward through the water column over a period of decades. The coral does not experience the change as an emergency. It grows, or it does not. Its polyps feed, or they stop. The reef structure maintains itself, or it begins to weaken. All of this happens slowly, in the dark, unobserved, in a place where no monitoring program currently operates with the continuity and temporal resolution to detect the change as it occurs. The ocean holds these processes in the same patient dark that it holds everything else. The change does not announce itself. The consequences do not come all at once. They accumulate slowly, in a pattern visible only across spans of time longer than most scientific programs and most human institutional memories. There is something to be said in this context for simply knowing what is there, for building the baseline inventory, the catalog of species, the map of communities, the record of chemical conditions, not because the inventory will prevent the change, but because the inventory is the minimum requirement for understanding what is being changed and at what rate. An ocean whose inhabitants are unnamed is an ocean whose losses cannot be counted. An ocean whose communities are unmapped is an ocean whose changes cannot be tracked. The survey is not a luxury. It is the precondition for any other understanding. The survey is proceeding. It is proceeding slowly, relative to the scale of the ocean, and it is proceeding faster than it has ever proceeded before, because the instruments are better and the international commitment is larger than at any previous point. The Seabed 2030 Initiative, the expansion of the ArgoFloat program to deep depths, the proliferation of autonomous underwater vehicles, The global-scale application of environmental DNA sampling. All of these are accelerating the rate at which the ocean's inventory is being established. And still, every expedition that goes somewhere new finds things that were not expected. Every new analytical technique applied to existing collections finds biology that the previous techniques missed. Every new sampling location adds species to the global list. The rate of discovery is not decreasing. The rate of the ocean's inventory being established is increasing. But the gap between the two remains enormous, and it will remain enormous for a very long time. The deep ocean has had a long time to accumulate what it contains. It will take a long time to catalog it. And in the interval, which is now, it holds what it holds in the dark and the quiet, neither revealed nor concealed, simply present in the full inventory of its existence. The mystery is not a barrier. It is an accurate description of the current relationship between human knowledge and the largest and deepest environment on the surface of the Earth. That relationship is changing, slowly, as everything in the deep ocean changes. But it is changing. And what has been found so far, the chemosynthetic communities, the deep biosphere, the lost city vents, the Vampiroteuthis, the barreli, the long-lived corals, the ancient Greenland sharks, the bioduck's patient revelation of its source, the Milky Sea glowing across 15,000 square kilometers visible from space. What has been found is remarkable enough to suggest that what remains unfound is remarkable too. The ocean does not disappoint when someone takes the time to look carefully. That has been true since the first rope was lowered over the side of the HMS Challenger in 1872. It remains true, and the rope is still going down, further each time, into darker water, finding things. The deep ocean and human perception of the deep ocean have always been separated by a gap that has nothing to do with scientific knowledge. The gap is experiential. No human senses the deep ocean in the way they sense a forest or a mountain or a coastal beach. There is no smell of deep seawater that a person knows from childhood. There is no sound of the deep ocean that travels through the air and arrives at a human ear. The deep ocean is known only through instruments and through the reports of those instruments, which translate the ocean into numbers and images and samples that can be carried back to the surface and examined. This is different from the way humans know most of the environment they inhabit. A naturalist who has spent years in a particular forest develops a felt sense of the forest, the way the light changes at different times of year, the particular calls of particular birds in particular seasons, the subtle shifts in humidity and temperature that indicate weather changes. This felt sense accumulates from direct sensory experience over time and allows a kind of intuitive understanding that supplements and sometimes exceeds what the formal scientific record contains. The history of naturalist science is full of observations that originated in this intuitive, sensory familiarity with a place and that subsequently proved to be scientifically significant. The deep ocean permits no such intuitive familiarity. The few people who have descended into the deep ocean in submersibles have described experiences of spatial disorientation, of the difficulty of maintaining a sense of scale in an environment with few reference points, of the way the relationship between the animal and the instrument, seeing the deep ocean through windows and cameras rather than with unmediated senses, creates a distance that is not only physical. Sylvia Earle, who has spent more time in deep-sea submersibles than almost any other scientist, has described the experience of the deep ocean as profoundly alien even after decades of familiarity with it. The alien quality does not diminish with repetition. The deep ocean is not a place that humans become accustomed to in the way they become accustomed to any terrestrial environment. This means that the database of informal, observational, felt knowledge that exists for forests and mountains and coastal seas, the accumulated observations of generations of naturalists and farmers and fishermen who lived close to a particular environment and knew it in the way you know something you have been around your whole life, does not exist for the deep ocean. There is no traditional ecological knowledge of the abyssal plain. There is no generational understanding of the bathypelagic zone that a community of deep-sea inhabitants has passed down. The deep ocean is known only from the outside through instruments by people who descend briefly and then return. The consequence is a specific kind of blindness. Human judgment about what is unusual requires a baseline of the normal. A bird watcher notices the unusual bird because they know what birds are normally present. A farmer notices the unusual behavior of the soil because they know how the soil normally looks and smells. The submersible pilot descending to a seamount at 1,000 meters depth is not sure what is normal for that seamount because no one has been there before and there is no prior baseline of normal. Everything is potentially unusual. Nothing is familiar enough to provide a reference for the unusual. This problem is being addressed in part by the development of long-term monitoring stations at specific deep-sea locations. A small number of deep-sea sites are instrumented with bottom-mounted sensors, time-lapse cameras, and acoustic instruments that record conditions continuously over months to years. The Neptune-cabled observatory network off the coast of British Columbia and the EMSO network in the European Atlantic and Mediterranean are examples of infrastructure designed to provide the continuous temporal coverage that episodic expeditions cannot. Over years of continuous recording, these systems are beginning to reveal what the normal looks like at the specific locations they monitor, what the seasonal variation in the biological community looks like, what the typical particle flux from above looks like, how the temperature and oxygen vary with depth and time. The picture emerging from these continuous records is more dynamic than the pre-monitoring view suggested. The deep sea was long assumed to be a stable environment, changing very slowly on geological timescales and barely at all on seasonal or annual timescales. The continuous records show otherwise. The benthic community at monitored sites in the North Atlantic and North Pacific shows marked seasonal variation in abundance and composition, driven by the arrival of food pulses from seasonal blooms at the surface above. A bloom of phytoplankton at the surface in spring produces a pulse of sinking organic material, phytodetritus, fecal pellets, marine snow, that arrives at the abyssal floor weeks to months later, triggering a measurable increase in biological activity. More organisms, more movement, more feeding, more reproduction. The deep ocean is not disconnected from the surface ocean's seasonal rhythms. It receives them, delayed and attenuated, through the medium of the biological pump. And the organisms of the deep sea respond to these pulses in ways that are only now being documented continuously enough to reveal the patterns. Before the continuous monitoring stations, the episodic expeditions that visited a site once every few years could not distinguish seasonal variation from long-term change. A site visited in spring would be compared with data from a visit the following autumn, and the difference might be attributed to environmental change rather than seasonal cycle. The monitoring stations resolve this ambiguity by being there continuously. What the monitoring stations cannot do is provide spatial coverage. Each station monitors one location. The deep ocean has many locations, and the variation between them is not yet characterized well enough to know how representative any single monitoring site is of the broader region. The Porcupine Abyssal Plain Observatory in the Northeast Atlantic has been monitoring one location on the abyssal plain since the 1990s and has produced invaluable long-term data. But the abyssal plain extends for thousands of kilometers in every direction from that station. Whether the dynamics observed at the station are representative of the broader plain, or whether they are specific to the particular characteristics of that location, is not established. The problem of spatial coverage in the deep ocean is the same problem as the problem of temporal coverage, expressed differently. There is not enough coverage, spatial or temporal, to characterize the deep ocean at the resolution needed to understand it as a dynamic system rather than a static snapshot. What there is, instead, is a growing collection of high-resolution windows into specific places and specific times, combined with the low-resolution global picture provided by satellite gravity data and sparse ship tracks. Between the high-resolution windows and the low-resolution global picture is a large intermediate scale, the scale of hundreds of kilometers, months to years, where the deep ocean is largely uncharacterized. The dynamics at this intermediate scale are where much of the ecologically and biogeochemically significant action occurs, and they remain mostly unknown. The ocean is patient about its dynamics. The eddies that carry water with specific temperature and chemistry from one part of the ocean to another, the internal waves that propagate through the deep ocean and mix different water masses, the deep bottom currents that carry Antarctic bottom water northward across the abyssal plains of the Atlantic. These processes operate on timescales of weeks to months and spatial scales of hundreds to thousands of kilometers. They are captured partially in the data from research cruises, partially in the records from moored instruments, partially in the output of ocean circulation models. The true three-dimensional, time-varying picture of these processes across the global deep ocean does not exist in any observational dataset. The deep ocean circulation is a fact. Its specifics are models, constrained by observations but not fully determined by them. The observation network is improving. The models are improving. The gap between what the observations show and what the models assume is closing. But it is not closed. And yet, within all of this incompleteness, within all of this limitation of coverage and resolution and duration, the deep ocean has revealed enough of itself to establish that it is one of the most remarkable environments on Earth. It supports life in forms that were not imagined before they were found. It operates on timescales and spatial scales that challenge any intuitive sense of what an environment can be. It maintains conditions that have persisted for millions of years and that have shaped organisms whose biology reflects that duration. Every question answered about the deep ocean opens additional questions. The discovery of hydrothermal vents opened questions about how the chemosynthetic communities are connected across hundreds of kilometers of bare seafloor between vent fields, how the larvae of tube worms find new vents to colonize, what dispersal mechanisms allow the vent community to persist and spread. Those questions are being studied and partially answered, and each answer produces new questions about the genetics of vent populations, the evolution of vent-specific adaptations, the chemical communication between organisms in the vent community. The questions proliferate at least as fast as the answers arrive. This is not a sign that the ocean is resistant to understanding. It is a sign that the ocean is genuinely complex, that understanding it is a project that will not be completed within any individual career or generation, and that each step of understanding reveals the full scope of what comes next. The deep ocean rewards patience of a particular kind, the patience to accept that the full picture will not arrive quickly, and that the partial picture accumulating from careful observation is genuinely illuminating even before it is complete. The observations continue, the instruments descend, the data arrives, and the ocean, vast and dark and very old, continues to hold what it holds, present and patient, in the deep water where no wind has ever blown and no light from any star has ever reached. The ocean's relationship with human knowledge is partly a story of tools and partly a story of questions. The tools have always been the limiting factor because the questions have been there since the first oceanographer wondered what was at the bottom of the sea. The tools available in 1872 when the Challenger expedition departed constrained the knowledge to what could be discovered by rope sounding and trawl net. The tools available in the 1930s constrained it to what could be sampled from ships or seen through the thick glass of a bathysphere window at the end of a cable. The tools available in the 1970s constrained it to what Alvin could reach and what acoustic surveys could profile. The tools available now, autonomous underwater vehicles capable of extended operation at full ocean depth, environmental DNA sampling from any water column depth, high-resolution multi-beam sonar on ships and AUVs, cabled seafloor observatories with real-time data transmission, machine learning algorithms that can analyze acoustic recordings and video footage at scales no human analyst could manage. These tools are an extraordinary advance over every previous generation. They are also clearly insufficient for the task. The ocean is too large, too deep, and too varied for any current combination of instruments to characterize it comprehensively. The instrument capability is approaching an inflection point. Autonomous platforms that can multiply the spatial coverage of ship-based research by factors of 10 or 100, combined with molecular methods that can inventory biodiversity without requiring the collection and identification of individual specimens. But the inflection point has not yet arrived. In the interval between the tools available and the tools needed lies the persistent unknown of the deep ocean. This interval is shortening. It will be shorter in 10 years than it is now, and shorter still in 20. The deep ocean at the end of the 21st century will be far better known than the deep ocean at the beginning. The surprises will still come. The ocean is too large and too varied for there to be no more surprises. But they will be surprises within a context of substantially greater baseline knowledge. What will those surprises be? The history of deep-sea exploration suggests that the most significant surprises will be in categories not currently recognized as categories. The chemosynthetic vent communities of 1977 were not on any list of possible discoveries, because the concept of a chemosynthetic ecosystem large enough to sustain macrofauna had not been formulated. The deep biosphere was not on any list, because subseafloor sediment was not conceptualized as a habitat. Lost City was not on any list, because alkaline serpentinization-driven hydrothermal systems at remote mid-ocean ridge locations were not a recognized category of investigation. The next major surprise, and there will be one because the rate of discovery has not decreased, will probably not be on the current list of suspected unknowns. It will be a new category, recognized only after the thing is found. The specifics of the deep-sea biological communities that have been found, the tube worm adaptations to vent fluid, the snailfish adaptations to hadal pressure, the Rimicaris shrimp's modified eye, suggest that the next generation of organism-specific discoveries will involve biology adapted to environmental conditions that have not been specifically sampled. There are hydrothermal vent systems in the deep ocean that have not been found yet. There are cold seep communities and ocean basins that have not been systematically surveyed. There are hadal trenches whose floor biology is known from a single sampling effort or not at all. Every one of these environments, when visited, will contain organisms adapted to those specific conditions, some of which will be new species and some of which will be new categories of adaptation. The pace of deep-sea biological discovery in the 21st century has already been remarkable. The description of new species from deep-sea collections, from museum archives, from new expeditions, from the analysis of preserved samples, has been occurring at rates of hundreds of new species per year in some taxonomic groups. The polychaete worms of the deep sea, the small crustaceans, the nematode worms of the sediment, the sponges, the soft corals, in every group that has been systematically studied with modern taxonomic methods, the proportion of new species in existing collections has been high. The species have been sitting in museum drawers and preservative jars for decades, collected during expeditions whose primary purpose was not biological survey, and the taxonomic expertise to analyze them has only recently been applied. This situation, organisms described from preserved material that was collected years or decades earlier, rather than from new expeditions, is characteristic of the current state of deep-sea taxonomy. There is more material in existing collections than there are taxonomists to analyze it. New expeditions are generating material faster than the analysis can proceed. The rate of formal species description is limited not by the availability of specimens, but by the availability of expert taxonomists with the time and resources to examine them. In several key deep-sea groups, the number of practicing taxonomists is small enough that the backlog of undescribed specimens grows faster than it can be cleared. This taxonomic bottleneck is a specific practical problem with a specific practical solution. More taxonomists, more funding for systematic biology, more time for the detailed comparative anatomy and genetic analysis that formal species description requires. The solution is not technically difficult. It is a matter of priority and resource allocation. The specimens exist. The methods exist. The expertise exists. Concentrated in a relatively small number of individuals at a relatively small number of institutions. Expanding the capacity would expand the rate of species description. Until that expansion occurs, the ocean continues to generate its surprises at a rate that outpaces the system designed to process them. This is not a crisis, but it is a bottleneck with consequences. Species that are described cannot be studied for behavior, ecology, or biochemistry until they are named and placed in the broader context of their phylogeny. Species sitting unnamed in museum jars are not accessible to the ecologist or the pharmacologist or the evolutionary biologist who might learn something important from them. The ocean's inventory is larger than the system for processing it. That has been true for a long time. It is true now, and it will remain true until either the inventory is substantially reduced, which is not going to happen, the ocean is not going to become simpler, or the processing system is substantially expanded. In the meantime, the inventory exists. Every organism in the deep ocean that has been collected and preserved is part of a record that can be examined when the resources to examine it are available. Every organism in the deep ocean that has not been collected is part of a record that is still being written. The record is ongoing. It is not static. The deep ocean does not stop generating new life, new adaptation, new variation, new biological innovation. It has been generating these things for 500 million years of complex multicellular life, and for billions of years before that. And the process has not slowed. The ocean is not a museum. It is not a record of what was. It is an ongoing process, continuous, evolving, adapting, changing in detail, while remaining recognizable in its broad character. Every organism in it is the current product of a continuous evolutionary lineage, reaching back to the origin of life. Every adaptation it possesses is the current solution to a problem that the environment has been posing in slightly varying form for the entirety of that lineage's history. The solutions are specific and intricate and beautiful in the way that anything refined over enormous amounts of time becomes beautiful. And they are, in large part, still unknown. Not hidden, not lost. Present in the dark water as they have always been. Waiting as the ocean waits, without impatience, as patient as the deep water itself, as quiet as the sediment on the abyssal plain, as steady as the cold, and very much alive. Consider what the deep ocean means as a presence on this planet, in the specific sense of its physical existence as the dominant terrain of the Earth's surface. The ocean covers 71% of the planet. Of that, the deep ocean, everything below the continental shelves, below 200 meters, covers the vast majority. The abyssal plains alone cover roughly 54% of the Earth's surface area. They are the most extensive landscape on the planet, bar none. They exceed the total land area of the Earth, and they are almost entirely unmapped at meaningful resolution and almost entirely unvisited. The abyssal plains are pale, flat, featureless at the scale of satellite observation, and extraordinarily complex at the scale of a square meter of seafloor. At the scale of a square meter, the abyssal sediment is a landscape of foraminiferous shells and mineral grains and organic aggregates and burrow openings and mound structures built by organisms living in and on the sediment. The diversity of organisms in that square meter, the polychaetes, the nematodes, the bacteria and archaea, the small crustaceans, is not well characterized at any location on the abyssal plain. The number of species that can coexist in a square meter of abyssal sediment is not known with confidence. The ecological relationships among those species are largely unstudied. Scale up from 1 square meter to 1 square kilometer of abyssal plain, and you have a landscape whose biological characterization would require, at minimum, dozens of sediment cores and epibenthic sleds, analyzed by multiple specialists over years of work. No such characterization has been done at any single square kilometer of the abyssal plain, at the level of rigor that a comparable square kilometer of, say, temperate forest or tropical reef has been characterized. The abyssal plain remains, in the most literal sense, one of the least known environments on the surface of the Earth. The depth at which the abyssal plain sits, typically 3,000 to 6,000 meters, places it below the reach of daylight, below the reach of significant surface ocean seasonal variation, except through the attenuated signal of the biological pump, and below the reach of most current research platforms. The ROV technology exists to reach it. The ships to deploy that technology exist. The scientific expertise to analyze what is found exists. What is lacking, mostly, is the funding and ship time to conduct systematic surveys at the spatial density needed to characterize the community. The abyssal plain is not exciting in the way that hydrothermal vents are exciting. It does not have the dramatic thermal and chemical gradients of the vent environment or the dense biological communities sustained by those gradients. It is, in the popular imagination, a featureless plain of mud extending in all directions in the cold dark. This is approximately correct at the scale of visual observation from a submersible or camera system, where the primary impression is of pale sediment, occasional tracks, rare large organisms separated by wide expanses of apparently empty seafloor. But the ecological reality of the abyssal plain is more complex than the visual impression suggests. The density of organisms per square meter is low compared to the shallow seafloor, but the total area is so large that the total number of organisms is enormous. The biomass of nematodes alone in the abyssal plain sediment, the tiny worms that live at densities of millions per square meter even at abyssal depths, exceeds the biomass of many more visible components of the abyssal community. The microbial biomass of the abyssal sediment is larger still. The ecological processes occurring in the sediment, decomposition, nutrient cycling, bioturbation by burrowing organisms, affect the chemistry of the overlying water and contribute to the global cycling of carbon and nutrients in ways that have implications for the whole ocean. The abyssal plain is not an inert substrate. It is an active ecological system. Its activity is slow and its participants are small, but its scale ensures that the sum of its small, slow activities is large. The ocean keeps this accounting, as it keeps all its accountings, without any requirement for external observation. The organisms do what they do in the dark and the cold. The chemistry evolves as it evolves. The record accumulates in the sediment. And the mystery persists. What is in the sediment? What does it do? What has it done over the long history of the abyssal plain? And what will it do as the conditions at the surface continue to change and the signals of those changes propagate downward through the water column into the place where the sediment meets the water? These questions are not rhetorical. They have real answers that are in principle discoverable by the methods available to deep-sea science. They simply have not been discovered yet, because the resources allocated to their discovery have not been sufficient to the scale of the task. The task is large. The ocean is large. It is the largest habitable space on the surface of the Earth by a wide margin, and it is the least known. This is not going to change quickly. The gap between what is known and what exists to be known will close, but slowly, over decades and centuries, as each generation of instruments and scientists adds to the record. There is a comfort in this if you are willing to find it. The ocean is not finished. It is not done with its surprises. It is not close to being fully understood. The discoveries that lie ahead, the species not yet found, the communities not yet visited, the biochemistries not yet analyzed, the sounds not yet identified, the evolutionary histories not yet traced, are as real as the discoveries that have already been made. They are out there, in the dark water, part of the ocean's inventory, patient and present and waiting to be found. The ocean does not know it is being studied. It does not know it is being mapped. It does not know about the instruments descending through its water column or the cameras approaching its floor. It simply is, in its entirety, at every moment, doing what it has always done. That indifference is not emptiness. It is the character of a very large and very old environment that has no need of observation to exist. It exists, it is full, and it is still, in most of what it contains, completely unknown. That is not a failure. It is an invitation. For whoever is willing to go, for whoever has the patience, for whoever can wait as long as it takes. The ocean will still be there. It will still be full. And it will still have things to show. The darkness of the deep ocean is not like any other darkness. It is a darkness that has existed at depth, without interruption, since the ocean formed on this planet. No geological event since the formation of the ocean basins has carried light to the abyssal floor. The darkness is ancient. The organisms living in it are adapted to it in ways that took millions of years to refine. The fish that produce light are producing it against that ancient darkness in exactly the way they have been producing it for whatever span of millions of years their lineage has occupied the deep. The bioluminescence is not alien to the darkness, it is part of the darkness. It emerged from it. There is no sharp boundary where the unknown begins. The ocean shades from the relatively familiar waters of the continental shelf, still largely unsurveyed in biological detail, through the twilight zone of the mesopelagic, known in outline, through the bathypelagic and the abyssopelagic and the hadal, each zone less known than the one above it. The unknown deepens with depth. It does not begin at a specific latitude or longitude or depth. It begins at the boundary of the last careful observation and extends from there in all directions. That boundary is much closer to the surface than most people imagine, and what lies beyond it is much larger than most people know. The ocean continues. It has always continued.